Mitochondrion. 101 languages. Mitochondrion. 2 mitochondria from mammalian lung tissue, displaying their matrix and membranes as shown by electron microscopy. Mitochondria redirects here. For the song by Kenichi Suzumiura, see Mitochondria, song. For the Canadian band, see Mitochondrion, band. A mitochondrion, PL mitochondria, is an organelle found in the cells of most eukaryotes, such as animals, plants and fungi. Mitochondria have a double membrane structure and use aerobic respiration to generate adenosine triphosphate, ATP, which is used throughout the cell as a source of chemical energy. 2. They were discovered by Albert von Kolliker in 1857. 3. In the voluntary muscles of insects. Meaning a thread-like granule, the term mitochondrion was coined by Carl Bender in 1898. The mitochondrion is popularly nicknamed the powerhouse of the cell, a phrase popularized by Philip Siekewitz in a 1957 Scientific American article of the same name. 4. Some cells in some multicellular organisms lack mitochondria, for example, mature mammalian red blood cells. The multicellular animal Hennegia salminicola is known to have retained mitochondrion-related organelles, despite a complete loss of their mitochondrial genome. 5, 6, 7. A large number of unicellular organisms, such as microsporidia, parabasalids and diplomonids, have reduced or transformed their mitochondria into other structures. 8. For example hydrogenosomes and mitosomes. 9. The oxymonids. Problems donating. Other ways to give frequently asked questions. We never sell your information. By submitting, you are agreeing to our donor privacy policy. The Wikimedia Foundation is a non-profit, tax-exempt organization. If you make a recurring donation, you will be debited by the Wikimedia Foundation until you notify us to stop. We'll send you an email, which will include a link to easy cancellation instructions. I already donated. How often would you like to donate? One time give monthly. Please select an amount, USD. The average donation in the United States is around $13. $2.75, $10, $15, $25, $50, $75, $100 other. Please select a payment method. Bank transfer, ACH. Maybe later. 1. Suzo RCN. 2. 3. Continue. Wikipedia still can't be sold. The 17th of October, an important update for readers in the United States. You deserve an explanation, so please don't skip this 1-minute read. We're sorry to interrupt, but it's Thursday, the 17th of October, and this message will be up for only a few hours. We ask you to react on the number of times you visited Wikipedia in the past year, and if you're able to give $2.75 to the Wikimedia Foundation. If everyone reading this gave just $2.75, we'd hit our goal in a few hours. In the age of AI, access to variable facts is crucial. Wikipedia is at the heart of online information, powering everything from your personal searches to emerging AI technologies. Your gift strengthens the knowledge of today and tomorrow. Just 2% of our readers donate, so if you have given in the past and Wikipedia still provides you with $2.75 worth of knowledge. Kindly donate today. If you are undecided, remember that any contribution helps, whether it's $2.75 or $25. Proud host of Wikipedia and its sister sites. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Diagram of animal mitochondrion. Details. Pronunciation /Mitochondrion /1. Part of cell. Identifiers. Latin organella. Mesh D008928. FMA 63835. Anatomical terms of microanatomy. Edit on Wikidata. Cell biology. Animal cell diagram. Components of a typical animal cell. 1 nucleolus. 2 nucleus. 3 ribosome, dots as part of 5. 4 vesicle. 5 rough endoplasmic reticulum. 6 Golgi apparatus, or Golgi body. 7 cytoskeleton. 8 smooth endoplasmic reticulum. 9 mitochondrion. 10 vacuole. 11 cytosol, fluid that contains organelles, with which comprises cytoplasm. 12 lysosome. 13 centrosome. 14 cell membrane. Cell biology. Mitochondrion. Monocercomonoids, streblomastics, and blattamonas have completely lost their mitochondria. 5, 10. Mitochondria are commonly between 0.75 and 3 micrometers 2 in cross-section, 11, but vary considerably in size and structure. Unless specifically stained, they are not visible. In addition to supplying cellular energy, mitochondria are involved in other tasks, such as signaling, cellular differentiation, and cell death, as well as maintaining control of the cell cycle and cell growth. 12. Mitochondrial biogenesis is in turn temporally coordinated with these cellular processes. 13, 14. Mitochondria have been implicated in several human disorders and conditions, such as mitochondrial diseases. 15. Cardiac dysfunction. 16. Heart failure. 17. And autism. 18. The number of mitochondria in a cell can vary widely by organism, tissue, and cell type. A mature red blood cell has no mitochondria, 19, whereas a liver cell can have more than 2,000. 20, 21. The mitochondrion is composed of compartments that carry out specialized functions. These compartments or regions include the outer membrane, intermembrane space, inner membrane, crista, and matrix. Although most of a eukaryotic cell's DNA is contained in the cell nucleus, the mitochondrion has its own genome, mitogenome, that is substantially similar to bacterial genomes. 22. This finding has led to general acceptance of the endosymbiotic hypothesis, that free-living prokaryotic ancestors of modern mitochondria permanently fused with eukaryotic cells in the distant past, evolving such that modern animals, plants, fungi, and other eukaryotes are able to respire to generate cellular energy. 23. Structure. Mitochondria may have a number of different shapes. 24. A mitochondrion contains outer and inner membranes composed of phospholipid bilayers and proteins. 20. The two membranes have different properties. Because of this, Double-membraned organization. There are 5 distinct parts to a mitochondrion. 1. The outer mitochondrial membrane. 2. The intermembrane space, the space between the outer and inner membranes. 3. The inner mitochondrial membrane. 4. The crystal space, formed by infoldings of the inner membrane, and 5. The matrix, space within the inner membrane, which is a fluid. Mitochondria have folding to increase surface area, which in turn increases ATP, adenosine triphosphate, production. Mitochondria stripped of their outer membrane are called mitoplasts. Outer membrane. The outer mitochondrial membrane, which encloses the entire organelle, is 60 to 75 angstroms, thick. It has a protein to phospholipid ratio similar to. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Components of a typical mitochondrion. 1. Outer membrane. 1.1 Porin. 2. Intermembrane space. 2.1 Intracrystal space. 2.2 Peripheral space. 3. Lamella. 3.1 Inner membrane. 3.11 Inner boundary membrane. 3.12 Crystal membrane. 3.2 Matrix. 3.3 Crista. 4 Mitochondrial DNA. 5 Matrix granule. 6 Ribosome. 7 ATP synthase. That of the cell membrane, about 1:1 by weight. It contains large numbers of integral membrane proteins called porins. A major trafficking protein is the pore-forming voltage-dependent ion channel, VDAC. The VDAC is the primary transporter of nucleotides, ions and metabolites between the cytosol and the intermembrane space. 25, 26. It is formed as a beta barrel that spans the outer membrane, similar to that in the gram-negative bacterial outer membrane. 27. Larger proteins can enter the mitochondrion if a signaling sequence at their N-terminus binds to a large multi-subunit protein called translocase in the outer membrane, which then actively moves them across the membrane. 28. Mitochondrial proteins are imported through specialized translocation complexes. The outer membrane also contains enzymes involved in such diverse activities as the elongation of fatty acids, oxidation of epinephrine, and the degradation of tryptophan. These enzymes include monoamine oxidase, retenan, insensitive NAD cytochrome C reductase, kynurenine hydroxylase and fatty acid CoA ligase. Disruption of the outer membrane permits proteins in the intermembrane space to leak into the cytosol, leading to cell death. 29. The outer mitochondrial membrane can associate with the endoplasmic reticulum, a membrane, in a structure called MAM, mitochondria-associated membrane. This is important in the mitochondria calcium signaling and is involved in the transfer of lipids between the and mitochondria. 30. Outside the outer membrane are small, diameter, 60 particles named subunits of Parson. Intermembrane space. The mitochondrial intermembrane space is the space between the outer membrane and the inner membrane. It is also known as perimitochondrial space. Because the outer membrane is freely permeable to small molecules, the concentrations of small molecules, such as ions and sugars, in the intermembrane space is the same as in the cytosol. 20. However, large proteins must have a specific signaling sequence to be transported across the outer membrane, so the protein composition of this space is different from the protein composition of the cytosol. One protein that is localized to the intermembrane space in this way is cytochrome C. 29. Inner membrane. Main article, inner mitochondrial membrane. The inner mitochondrial membrane contains proteins with three types of functions. 20. 1. Those that perform the electron transport chain redox reactions. 2. ATP synthase, which generates ATP in the matrix. 3. Specific transport proteins that regulate metabolite passage into and out of the mitochondrial matrix. It contains more than 151 different polypeptides, and has a very high protein to phospholipid ratio, more than 3:1 by weight, which is about 1 protein for 15 phospholipids. The inner membrane is home to around 1/5 of the total protein in a mitochondrion. 31. Additionally, the inner membrane is rich in an unusual phospholipid, cardiolipin. This phospholipid was originally discovered in cow hearts in 1942, and is usually characteristic of mitochondrial and bacterial plasma membranes. 32. Cardiolipin contains 4 fatty acids rather than 2, and may help to make the inner membrane impermeable. 20. And its disruption can lead to multiple clinical disorders including neurological disorders and cancer. 33. Unlike the outer membrane, the inner membrane does not contain porins, and is highly impermeable to all molecules. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Cross-sectional image of Crista in a rat liver mitochondrion to demonstrate the likely 3D structure and relationship to the inner membrane. Almost all ions and molecules require special membrane transporters to enter or exit the matrix. Proteins are ferried into the matrix via the translocase of the inner membrane, TIM, complex or via OXA1L. 28. In addition, there is a membrane potential across the inner membrane, formed by the action of the enzymes of the electron transport chain. Inner membrane fusion is mediated by the inner membrane protein OPA1. 34. Crista. Main article, Crista. The inner mitochondrial membrane is compartmentalized into numerous folds, called crista, which expand the surface area of the inner mitochondrial membrane, enhancing its ability to produce ATP. For typical liver mitochondria, the area of the inner membrane is about 5 times as large as that of the outer membrane. This ratio is variable, and mitochondria from cells that have a greater demand for ATP, such as muscle cells, contain even more crista. Mitochondria within the same cell can have substantially different crista density, with the ones that are required to produce more energy having much more crista membrane surface. 35. These folds are studded with small round bodies known as F1 particles or oxysomes. 36. Matrix. Main article, mitochondrial matrix. The matrix is the space enclosed by the inner membrane. It contains about 2/3 of the total proteins in a mitochondrion. 20. The matrix is important in the production of ATP with the aid of the ATP synthase contained in the inner membrane. The matrix contains a highly concentrated mixture of hundreds of enzymes, special mitochondrial ribosomes, tRNA, and several copies of the mitochondrial DNA genome. Of the enzymes, the major functions include oxidation of pyruvate and fatty acids, and the citric acid cycle. 20. The DNA molecules are packaged into nucleotides by proteins, one of which is TFAM. 37. Function. The most prominent roles of mitochondria are to produce the energy currency of the cell, ATP, i.e., phosphorylation of ADP, through respiration, and to regulate cellular metabolism. 21. The central set of reactions involved in ATP production are collectively known as the citric acid cycle, or the Krebs cycle, and oxidative phosphorylation. However, the mitochondrion has many other functions in addition to the production of ATP. Energy conversion. A dominant role for the mitochondria is the production of ATP, as reflected by the large number of proteins in the inner membrane for this task. This is done by oxidizing the major products of glucose, pyruvate, and NAD, which are produced in the cytosol. 21. This type of cellular respiration, known as aerobic respiration, is dependent on the presence of oxygen. When oxygen is limited, the glycolytic products will be metabolized by anaerobic fermentation, a process that is independent of the mitochondria. 21. The production of ATP from glucose and oxygen has an approximately 13 times higher yield during aerobic respiration compared to fermentation. 38. Plant mitochondria can also produce a limited amount of ATP, either by breaking the sugar produced during photosynthesis or without oxygen by using the alternate substrate nitrite. 39. ATP crosses out through the inner membrane with the help of a specific protein, and across the outer membrane via porins. 40. After conversion of ATP to ADP by dephosphorylation that releases energy, ADP returns via the same route. Pyruvate and the citric acid cycle. Main article, Citric acid cycle. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Electron transport chain in the mitochondrial intermembrane space. Pyruvate molecules produced by glycolysis are actively transported across the inner mitochondrial membrane, and into the matrix, where they can either be oxidized and combined with coenzyme to form CoA, acetyl-CoA, and NAD, 21, or they can be carboxylated by pyruvate carboxylase to form oxalostate. This latter reaction fills up the amount of oxalostate in the citric acid cycle and is therefore an anaplerotic reaction, increasing the cycle's capacity to metabolize acetyl-CoA when the tissue's energy needs, for example, in muscle, are suddenly increased by activity. 41. In the citric acid cycle, all the intermediates, for example citrate, isocitrate, alpha-ketoglutarate, succinate, fumarate, malate and oxalostate, are regenerated during each turn of the cycle. Adding more of any of these intermediates to the mitochondrion therefore means that the additional amount is retained within the cycle, increasing all the other intermediates as one is converted into the other. Hence, the addition of any one of them to the cycle has an anaplerotic effect, and its removal has a cataplerotic effect. These anaplerotic and cataplerotic reactions will, during the course of the cycle, increase or decrease the amount of oxalostate available to combine with acetyl-CoA to form citric acid. This in turn increases or decreases the rate of ATP production by the mitochondrion, and thus the availability of ATP to the cell. 41. Acetyl-CoA, on the other hand, derived from pyruvate oxidation, or from the beta oxidation of fatty acids, is the only fuel to enter the citric acid cycle. With each turn of the cycle, one molecule of acetyl-CoA is consumed for every molecule of oxalostate present in the mitochondrial matrix, and is never regenerated. It is the oxidation of the acetate portion of acetyl-CoA that produces CO2 and water, with the energy thus released captured in the form of ATP. 41. In the liver, the carboxylation of cytosolic pyruvate into intramitochondrial oxalostate is an early step in the gluconeogenic pathway, which converts lactate and deaminated alanine into glucose. 21, 41, under the influence of high levels of glucagon and / or epinephrine in the blood. 41. Here, the addition of oxalostate to the mitochondrion does not have a net anaplerotic effect, as another citric acid cycle intermediate, malate, is immediately removed from the mitochondrion to be converted to cytosolic oxalostate, and ultimately to glucose, in a process that is almost the reverse of glycolysis. 41. The enzymes of the citric acid cycle are located in the mitochondrial matrix, with the exception of succinate dehydrogenase, which is bound to the inner mitochondrial membrane as part of complex 2. 42. The citric acid cycle oxidizes the acetyl-CoA to carbon dioxide, and, in the process, produces reduced cofactors, 3 molecules of NAD and 1 molecule of FAD. 2. That are a source of electrons for the electron transport chain, and a molecule of GTP, which is readily converted to an ATP. 21. O2 and NAD, energy-releasing reactions. Main articles, oxidative phosphorylation and electron transport chain. The electrons from NAD and FAD 2 are transferred to oxygen, O2, and hydrogen, protons, in several steps via an electron transport chain. NAD and FAD 2 molecules are produced within the matrix via the citric acid cycle and in the cytoplasm by glycolysis. Reducing equivalents from the cytoplasm can be imported via the malate aspartate shuttle system of antiporter proteins or fed into the electron transport chain using a glycerol phosphate shuttle. 21. The major energy-releasing reactions, 43, 44, that make the mitochondrion the powerhouse of the cell occur at protein complexes I, 3 and 4 in the inner mitochondrial membrane. NAD dehydrogenase, ubiquinone, cytochrome C reductase, and cytochrome C oxidase. At complex 4, O2 reacts with the reduced form of iron in cytochrome C. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Releasing a lot of free energy, 44, 43, from the reactants without breaking bonds of an organic fuel. The free energy put into remove an electron from Fe2+ is released at complex 3 when Fe3+ of cytochrome C reacts to oxidize ubiquinol, QH. 2. Sad smiley. The ubiquinone, Q, generated reacts, in complex I, with NAD. While the reactions are controlled by an electron transport chain, free electrons are not amongst the reactants or products in the 3 reactions shown and therefore do not affect the free energy released, which is used to pump protons, H+, into the intermembrane space. This process is efficient, but a small percentage of electrons may prematurely reduce oxygen, forming reactive oxygen species such as superoxide. 21. This can cause oxidative stress in the mitochondria and may contribute to the decline in mitochondrial function associated with aging. 45. As the proton concentration increases in the intermembrane space, a strong electrochemical gradient is established across the inner membrane. The protons can return to the matrix through the ATP synthase complex, and their potential energy is used to synthesize ATP from ADP and inorganic phosphate, P. I. 21. This process is called chemiosmosis, and was first described by Peter Mitchell, 46-47, who was awarded the 1978 Nobel Prize in Chemistry for his work. Later, part of the 1997 Nobel Prize in Chemistry was awarded to Paul D. Boyer and John E. Walker for their clarification of the working mechanism of ATP synthase. 48. Heat production. Under certain conditions, protons can re-enter the mitochondrial matrix without contributing to ATP synthesis. This process is known as proton leak or mitochondrial uncoupling and is due to the facilitated diffusion of protons into the matrix. The process results in the unharnessed potential energy of the proton electrochemical gradient being released as heat. 21. The process is mediated by a proton channel called thermogenin, or UCP1. 49. Thermogenin is primarily found in brown adipose tissue, or brown fat, and is responsible for non-shivering thermogenesis. Brown adipose tissue is found in mammals, and is at its highest levels in early life and in hibernating animals. In humans, brown adipose tissue is present at birth and decreases with age. 49. Mitochondrial fatty acid synthesis. Mitochondrial fatty acid synthesis, MFASI, is essential for cellular respiration and mitochondrial biogenesis. 50. It is also thought to play a role as a mediator in intracellular signaling due to its influence on the levels of bioactive lipids, such as lysophospholipids and sphingolipids. 51. Octanoyl ACP, C8, is considered to be the most important end product of MFASI, which also forms the starting substrate of lipoic acid biosynthesis. 52. Since lipoic acid is the cofactor of important mitochondrial enzyme complexes, such as the pyruvate dehydrogenase complex, PDC, ketoglutarate dehydrogenase complex, OGDC, branched chain peptide dehydrogenase complex, BCKDC, and in the glycine cleavage system, GCS, MFASI has an influence on energy metabolism. 53. Other products of MFASI play a role in the regulation of mitochondrial translation, FES cluster biogenesis and assembly of oxidative phosphorylation complexes. 52. Furthermore, with the help of MFASI and acylated ACP, acetyl co-regulates its consumption in mitochondria. 52. Uptake, storage and release of calcium ions. The concentrations of free calcium in the cell can regulate an array of reactions and is important for signal transduction in the cell. Mitochondria can transiently store calcium, a contributing process for the cell's homeostasis of calcium. 54-55. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Transmission electron micrograph of a chondrocyte, stained for calcium, showing its nucleus, N, and mitochondria, M. Their ability to rapidly take in calcium for later release makes them good cytosolic buffers for calcium. 56, 57, 58. The endoplasmic reticulum, is the most significant storage site of calcium. 59, and there is a significant interplay between the mitochondrion and with regard to calcium. 60. The calcium is taken up into the matrix by the mitochondrial calcium uniporter on the inner mitochondrial membrane. 61. It is primarily driven by the mitochondrial membrane potential. 55. Release of this calcium back into the cells interior can occur via a sodium-calcium exchange protein or via calcium-induced calcium release pathways. 61. This can initiate calcium spikes or calcium waves with large changes in the membrane potential. These can activate a series of second messenger system proteins that can coordinate processes such as neurotransmitter release in nerve cells and release of hormones in endocrine cells. 62. Ca2+ influx to the mitochondrial matrix has recently been implicated as a mechanism to regulate respiratory bioenergetics by allowing the electrochemical potential across the membrane to transiently pulse from dominated to pH-dominated, facilitating a reduction of oxidative stress. 63. In neurons, concomitant increases in cytosolic and mitochondrial calcium act to synchronize neuronal activity with mitochondrial energy metabolism. Mitochondrial matrix calcium levels can reach the tens of micromolar levels, which is necessary for the activation of isocitrate dehydrogenase, one of the key regulatory enzymes of the Krebs cycle. 64. Cellular proliferation regulation. The relationship between cellular proliferation and mitochondria has been investigated. Tumor cells require ample ATP to synthesize bioactive compounds such as lipids, proteins, and nucleotides for rapid proliferation. 65. The majority of ATP in tumor cells is generated via the oxidative phosphorylation pathway, OXPHOS. 66. Interference with OXPHOS cause cell cycle arrest, suggesting that mitochondria play a role in cell proliferation. 66. Mitochondrial ATP production is also vital for cell division and differentiation in infection. 67. In addition to basic functions in the cell including the regulation of cell volume, solute concentration, and cellular architecture. 68, 69, 70. ATP levels differ at various stages of the cell cycle, suggesting that there is a relationship between the abundance of ATP and the cell's ability to enter a new cell cycle. 71. ATP's role in the basic functions of the cell make the cell cycle sensitive to changes in the availability of mitochondrial-derived ATP. 71. The variation in ATP levels at different stages of the cell cycle support the hypothesis that mitochondria play an important role in cell cycle regulation. 71. Although the specific mechanisms between mitochondria and the cell cycle regulation is not well understood, studies have shown that low-energy cell cycle checkpoints monitor the energy capability before committing to another round of cell division. 12. Programmed cell death and innate immunity. Further information: programmed cell death intrinsic pathway. Programmed cell death, PCD, is crucial for various physiological functions, including organ development and cellular homeostasis. It serves as an intrinsic mechanism to prevent malignant transformation and plays a fundamental role in immunity by aiding in antiviral defense, pathogen elimination, inflammation, and immune cell recruitment. 72. Mitochondria have long been recognized for their central role in the intrinsic pathway of apoptosis, a form of PCD. 73. In recent decades, they have also been identified as a signaling hub for much of the innate immune system. 74. The endosymbiotic origin of mitochondria distinguishes them from other cellular components, and the exposure of mitochondrial elements to the cytosol can trigger the same pathways as infection markers. These pathways lead to apoptosis, autophagy, or the induction of pro-inflammatory genes. 75, 74. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Mitochondria contribute to apoptosis by releasing cytochrome C, which directly induces the formation of apoptosomes. Additionally, they are a source of various damage-associated molecular patterns, DAMPs. These DAMPs are often recognized by the same pattern recognition receptors, PRs, that respond to pathogen-associated molecular patterns, PAMPs, during infections. 76. For example, mitochondrial mount DNA resembles bacterial DNA due to its lack of CpG methylation and can be detected by toll-like receptor 9 and CGAS. 77. Double-stranded RNA, dsRNA, produced due to bidirectional mitochondrial transcription, can activate viral sensing pathways through RIG-like receptors. 78. Additionally, the N-formylation of mitochondrial proteins, similar to that of bacterial proteins, can be recognized by formal peptide receptors. 79, 80. Normally, these mitochondrial components are sequestered from the rest of the cell but are released following mitochondrial membrane permeabilization during apoptosis or passively after mitochondrial damage. However, mitochondria also play an active role in innate immunity, releasing mount DNA in response to metabolic cues. 74. Mitochondria are also the localization site for immune and apoptosis regulatory proteins, such as BAX, MAVs, located on the outer membrane, and NLRX1, found in the matrix. These proteins are modulated by the mitochondrial metabolic status and mitochondrial dynamics. 74, 81, 82. Additional functions. Mitochondria play a central role in many other metabolic tasks, such as signaling through mitochondrial reactive oxygen species, 83. Regulation of the membrane potential, 21. Calcium signaling, including calcium-evoked apoptosis, 84. Regulation of cellular metabolism, 12. Certain heme synthesis reactions, 85, see also, porphyrin. Steroid synthesis, 56. Hormonal signaling, 86. Mitochondria are sensitive and responsive to hormones, in part by the action of mitochondrial estrogen receptors, MTERs. These receptors have been found in various tissues and cell types, including brain, 87, and heart, 88. Development and function of immune cells, 89. Neuronal mitochondria also contribute to cellular quality control by reporting neuronal status towards microglia through specialized somatic junctions. 90. Mitochondria of developing neurons contribute to intercellular signaling towards microglia, which communication is indispensable for proper regulation of brain development. 91. Some mitochondrial functions are performed only in specific types of cells. For example, mitochondria in liver cells contain enzymes that allow them to detoxify ammonia, a waste product of protein metabolism. A mutation in the genes regulating any of these functions can result in mitochondrial diseases. Mitochondrial proteins, proteins transcribed from mitochondrial DNA, vary depending on the tissue and the species. In humans, 615 distinct types of proteins have been identified from cardiac mitochondria, 92, whereas in rats, 940 proteins have been reported. 93. The mitochondrial proteome is thought to be dynamically regulated. 94. Organization and distribution. Mitochondria, or related structures, are found in all eukaryotes, except the oxymonid monocercomonoids. 5. Although commonly depicted as bean-like structures, they form a highly dynamic network in the majority of cells where they constantly undergo fission and fusion. The population of all the mitochondria of a given cell constitutes the chondriome. 95. Mitochondria vary in number and location according to cell type. A single mitochondrion is often found in unicellular organisms, while human liver cells have about 1,000–2,000 mitochondria per cell, making up 1/5 of the cell volume. 20. The mitochondrial content of otherwise similar cells can vary substantially in size and membrane potential. 96, with differences. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Typical mitochondrial network. Green, in 2 human cells, HeLa cells. Arising from sources including uneven partitioning at cell division, leading to extrinsic differences in ATP levels and downstream cellular processes. 97. The mitochondria can be found nestled between myofibrils of muscle or wrapped around the sperm flagellum. 20. Often, they form a complex 3D branching network inside the cell with the cytoskeleton. The association with the cytoskeleton determines mitochondrial shape, which can affect the function as well. 98. Different structures of the mitochondrial network may afford the population a variety of physical, chemical, and signaling advantages or disadvantages. 99. Mitochondria in cells are always distributed along microtubules, and the distribution of these organelles is also correlated with the endoplasmic reticulum. 100. Recent evidence suggests that vimentin, one of the components of the cytoskeleton, is also critical to the association with the cytoskeleton. 101. Mitochondria-associated membrane, MAM. Main article, Mitochondria-associated membranes. The mitochondria-associated membrane, MAM, is another structural element that is increasingly recognized for its critical role in cellular physiology and homeostasis. Once considered a technical snag in cell fractionation techniques, the alleged vesicle contaminants that invariably appeared in the mitochondrial fraction have been re-identified as membranous structures derived from the MAM, the interface between mitochondria and the 102. Physical coupling between these two organelles had previously been observed in electron micrographs and has more recently been probed with fluorescence microscopy. 102. Such studies estimate that at the MAM, which may comprise up to 20% of the mitochondrial outer membrane, the and mitochondria are separated by a mere 1025 nm and held together by protein tethering complexes. 102, 30, 103. Purified MAM from subcellular fractionation is enriched in enzymes involved in phospholipid exchange, in addition to channels associated with Ca2+ signaling. 102, 103. These hints of a prominent role for the MAM in the regulation of cellular lipid stores and signal transduction have been borne out, with significant implications for mitochondrial-associated cellular phenomena, as discussed below. Not only has the MAM provided insight into the mechanistic basis underlying such physiological processes as intrinsic apoptosis and the propagation of calcium signaling, but it also favors a more refined view of the mitochondria. Though often seen as static, isolated powerhouses hijacked for cellular metabolism through an ancient endosymbiotic event, the evolution of the MAM underscores the extent to which mitochondria have been integrated into overall cellular physiology, with intimate physical and functional coupling to the endomembrane system. Phospholipid transfer. The MAM is enriched in enzymes involved in lipid biosynthesis, such as phosphatidylserine synthase on the phase and phosphatidylserine decarboxylase on the mitochondrial phase. 104, 105. Because mitochondria are dynamic organelles, constantly undergoing fission and fusion events, they require a constant and well-regulated supply of phospholipids for membrane integrity. 106, 107. But mitochondria are not only a destination for the phospholipids they finish synthesis of. Rather, this organelle also plays a role in inter-organelle trafficking of the intermediates and products of phospholipid biosynthetic pathways, ceramide and cholesterol metabolism, and glycosphingolipid anabolism. 105, 107. Such trafficking capacity depends on the MAM, which has been shown to facilitate transfer of lipid intermediates between organelles. 104. In contrast to the standard vesicular mechanism of lipid transfer, evidence indicates that the physical proximity of the and mitochondrial membranes at the MAM allows for lipid flipping between opposed bilayers. 107. Despite this unusual and seemingly energetically unfavorable mechanism, such transport does not require ATP. 107. Instead, in yeast, it has been shown to be dependent on a multiprotein tethering structure termed the mitochondria. In counterstructure, ORMs, although it remains unclear whether this structure directly mediates lipid transfer or is required to keep the membranes in sufficiently close proximity to lower the energy barrier for lipid flipping. 107, 108. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The MAM may also be part of the secretory pathway, in addition to its role in intracellular lipid trafficking. In particular, the MAM appears to be an intermediate destination between the rougher and the Golgi in the pathway that leads to very low density lipoprotein, or VLDL, assembly and secretion. 105, 109. The MAM thus serves as a critical metabolic and trafficking hub in lipid metabolism. Calcium signaling. A critical role for the in calcium signaling was acknowledged before such a role for the mitochondria was widely accepted, in part because the low affinity of Ca2+ channels localized to the outer mitochondrial membrane seemed to contradict this organelle's purported responsiveness to changes in intracellular Ca2+ flux. 102, 59. But the presence of the MAM resolves this apparent contradiction. The close physical association between the two organelles results in Ca2+ Microdomains at contact points that facilitate efficient Ca2+ transmission from the to the mitochondria. 102. Transmission occurs in response to so-called Ca2+ puffs generated by spontaneous clustering and activation of IP3R, a canonical membrane Ca2+ channel. 102. 30. The fate of these puffs, in particular, whether they remain restricted to isolated locales or integrated into Ca2+ waves for propagation throughout the cell, is determined in large part by MAM dynamics. Although reuptake of Ca2+ by the concomitant with its release, modulates the intensity of the puffs, thus insulating mitochondria to a certain degree from high Ca2+ exposure, the MAM often serves as a firewall that essentially buffers Ca2+ puffs by acting as a sink into which free ions released into the cytosol can be funneled. 102, 110, 111. This Ca2+ tunneling occurs through the low affinity Ca2+ receptive DAC1, which recently has been shown to be physically tethered to the IP3R clusters on the membrane and enriched at the MAM. 102, 30, 112. The ability of mitochondria to serve as a Ca2+ sink is a result of the electrochemical gradient generated during oxidative phosphorylation, which makes tunneling of the cation an exergonic process. 112. Normal, mild calcium influx from cytosol into the mitochondrial matrix causes transient depolarization that is corrected by pumping out protons. But transmission of Ca2+ is not unidirectional, rather, it is a two-way street. 59. The properties of the Ca2+ pump circuit and the channel IP3 are present on the membrane facilitate feedback regulation coordinated by MAM function. In particular, the clearance of Ca2+ by the MAM allows for spatiotemporal patterning of Ca2+ signaling because Ca2+ alters IP3R activity in a biphasic manner. 102. Circuit is likewise affected by mitochondrial feedback. Uptake of Ca2+ by the MAM stimulates ATP production, thus providing energy that enables circuit to reload the with Ca2+ for continued Ca2+ efflux at the MAM. 110, 112. Thus, the MAM is not a passive buffer for Ca2+ puffs, rather it helps modulate further Ca2+ signaling through feedback loops that affect dynamics. Regulating release of Ca2+ at the MAM is especially critical because only a certain window of Ca2+ uptake sustains the mitochondria, and consequently the cell, at homeostasis. Sufficient intraorganelle Ca2+ signaling is required to stimulate metabolism by activating dehydrogenase enzymes critical to flux through the citric acid cycle. 113, 114. However, once Ca2+ signaling in the mitochondria passes a certain threshold, it stimulates the intrinsic pathway of apoptosis in part by collapsing the mitochondrial membrane potential required for metabolism. 102. Studies examining the role of pro- and anti-apoptotic factors support this model. For example, the anti-apoptotic factor BCL2 has been shown to interact with IP3RS to reduce Ca2+ filling of the leading to reduced efflux at the MAM and preventing collapse of the mitochondrial membrane. Potential post-apoptotic stimuli. 102. Given the need for such fine regulation of Ca2+ signaling, it is perhaps unsurprising that dysregulated mitochondrial Ca2+ has been implicated in several neurodegenerative diseases, while the catalogue of tumor Suppressors includes a few that are enriched at the MAM. 112. Molecular basis for tethering. Recent advances in the identification of the tethers between the mitochondrial and membranes suggest that the scaffolding function of the molecular elements involved is secondary to other, non-structural functions. In yeast, UMS, a multiprotein complex of interacting and mitochondrial resident membrane proteins, is required for lipid transfer at the MAM and exemplifies this principle. One of its components, for example, is also a constituent of the protein complex. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF model of the yeast multimeric tethering complex, UMS, required for insertion of transmembrane beta-barrel proteins into the lipid bilayer. 107, however, a homologue of the UMS complex has not yet been identified in mammalian cells. Other proteins implicated in scaffolding likewise have functions independent of structural tethering at the MAM, for example, resident and mitochondrial resident mitofusins form heterocomplexes that regulate the number of inter-organelle contact sites, although mitofusins were first identified for their role in fission and fusion events between individual mitochondria. 102. Glucose-related protein 75, GRP75, is another dual function protein. In addition to the matrix pool of GRP75, a portion serves as a chaperone that physically links the mitochondrial and Ca2+ channels of DAC and IP3R for efficient Ca2+ transmission at the MAM. 102, 30. Another potential tether is sigma-1R, a non-opioid receptor whose stabilization of a resident IP3R may preserve communication at the MAM during the metabolic stress response. 115, 116. Perspective. The MAM is a critical signaling, metabolic, and trafficking hub in the cell that allows for the integration of and mitochondrial physiology. Coupling between these organelles is not simply structural but functional as well and critical for overall cellular physiology and homeostasis. The MAM thus offers a perspective on mitochondria that diverges from the traditional view of this organelle as a static, isolated unit appropriated for its metabolic capacity by the cell. 117. Instead, this mitochondrial interface emphasizes the integration of the mitochondria, the product of an endosymbiotic event, into diverse cellular processes. Recently it has also been shown that mitochondria and MAMs in neurons are anchored to specialized intercellular communication sites, so-called somatic junctions. Microglial processes monitor and protect neuronal functions at these sites, and MAMs are supposed to have an important role in this type of cellular quality control. 90. Origin and evolution. Main article, Semiogenesis. There are two hypotheses about the origin of mitochondria, endosymbiotic and autogenous. The endosymbiotic hypothesis suggests that mitochondria were originally prokaryotic cells, capable of implementing oxidative mechanisms that were not possible for eukaryotic cells. They became endosymbionts living inside the eukaryote. 23, 118, 119, 120. In the autogenous hypothesis, mitochondria were born by splitting off a portion of DNA from the nucleus of the eukaryotic cell at the time of divergence with the prokaryotes. This DNA portion would have been enclosed by membranes, which could not be crossed by proteins. Since mitochondria have many features in common with bacteria, the endosymbiotic hypothesis is the more widely accepted of the two accounts. 120, 121. A mitochondrion contains DNA, which is organized as several copies of a single, usually circular chromosome. This mitochondrial chromosome contains genes for redox proteins, such as those of the respiratory chain. The core hypothesis proposes that this co-location is required for redox regulation. The mitochondrial genome codes for some RNAs of ribosomes, and the 22 tRNAs necessary for the translation of mRNAs into protein. The circular structure is also found in prokaryotes. The protomitochondrion was probably closely related to rickettsia. 122, 123. However, the exact relationship of the ancestor of mitochondria to the alpha proteobacteria and whether the mitochondrion was formed at the same time or after the nucleus remains controversial. 124. For example, it has been suggested that the SAR11 clade of bacteria shares a relatively recent common ancestor with the mitochondria. 125, while phylogenomic analyses indicate that mitochondria evolved from a pseudomonodonta lineage that is closely related to or a member of alpha proteobacteria. 126, 127. Some papers describe mitochondria as sister to the alpha proteobacteria, together forming the sister the Marinepproteo1 group, together forming the sister to Magnetococcidae. 128, 129, 130, 131. Proteobacteria. Alpha Proteobacteria SL. Magnetococcales. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Evolution of MROs. The circular 16569 BP human mitochondrial genome encoding 37 genes, i.e., 28 on the H strand and 9 on the L strand. Marinepproteo1. Mitochondria. Alpha Proteobacteria SS. The ribosomes coded for by the mitochondrial DNA are similar to those from bacteria in size and structure. 132. They closely resemble the bacterial 70S ribosome and not the 80S cytoplasmic ribosomes, which are coded for by nuclear DNA. The endosymbiotic relationship of mitochondria with their host cells was popularized by Lynn Margulies. 133. The endosymbiotic hypothesis suggests that mitochondria descended from aerobic bacteria that somehow survived endocytosis by another cell, and became incorporated into the cytoplasm. The ability of these bacteria to conduct respiration in host cells that had relied on glycolysis and fermentation would have provided a considerable evolutionary advantage. This symbiotic relationship probably developed 1.7 to 2 billion years ago. 134, 135. A few groups of unicellular eukaryotes have only vestigial mitochondria or derived structures, the microsporidians, metamonads, and archamoeba. 136. These groups appear as the most primitive eukaryotes on phylogenetic trees constructed using RNA information, which once suggested that they appeared before the origin of mitochondria. However, this is now known to be an artifact of long branch attraction. They are derived groups and retain genes or organelles derived from mitochondria. E.g., mitosomes and hydrogenosomes. 8. Hydrogenosomes, mitosomes, and related organelles as found in some Loricifera, e.g. Spiniloricus, 137, 138, and Mixozo, e.g. Hennegias are together classified as MROs, mitochondrion-related organelles. 7. 139. Monocercomonoids and other oxymonids appear to have lost their mitochondria completely, and at least some of the mitochondrial functions seem to be carried out by cytoplasmic proteins now. 5. 140, 10. Mitochondrial genetics. Main article, mitochondrial DNA. Mitochondria contain their own genome. The human mitochondrial genome is a circular double-stranded DNA molecule of about 16 kilobases. 141. It encodes 37 genes, 13 for subunits of respiratory complexes I, 3, 4 and V. 22 for mitochondrial tRNA, for the 20 standard amino acids, plus an extra gene for leucine and serine, and 2 for RNA, 12S and 16S RNA. 141. 1. Mitochondrion can contain 2 to 10 copies of its DNA. 142. One of the two mitochondrial DNA, mount DNA, strands has a disproportionately high ratio of the heavier nucleotides adenine and guanine, and this is termed the heavy strand, or H strand, whereas the other strand is termed the light strand, or L strand. The weight difference allows the two strands to be separated by centrifugation. Mount DNA has one long non-coding stretch known as the non-coding region, NCR, which contains the heavy strand promoter, HSP, and light strand promoter, LSP, for RNA transcription, the origin of replication for the H strand, or EH, localized on the L strand, 3 conserved sequence boxes, CSBs 1-3, and a termination-associated sequence, TARS. The origin of replication for the L strand, or EL, is localized on the H strand 11,000 BP downstream of OREH, located within a cluster of genes coding for tRNA. 143. As in prokaryotes, there is a very high proportion of coding DNA and an absence of repeats. Mitochondrial genes are transcribed as multigenic transcripts, which are cleaved and polyadenylated to yield mature mNAs. Most proteins necessary for mitochondrial function are encoded by genes in the cell nucleus, and the corresponding proteins are imported into the mitochondrion. 144. The exact number of genes encoded by the nucleus and the mitochondrial genome differs. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Exceptions to the standard genetic code in mitochondria, 20. Organism code on standard mitochondria. Mammals AGA, AGG arginine stop codon. Invertebrates AGA, AGG arginine serine. Fungi CUA leucine threonine. All of the above AUA isoleucine methionine. UGA stop codon tryptophan. Between species. Most mitochondrial genomes are circular. 145. In general, mitochondrial DNA lacks introns, as is the case in the human mitochondrial genome. 144. However, introns have been observed in some eukaryotic mitochondrial DNA. 146. Such as that of yeast. 147, and protists. 148, including Dictyostelium discoideum. 149. Between protein coding regions, tRNAs are present. Mitochondrial tRNA genes have different sequences from the nuclear tRNAs, but lookalikes of mitochondrial tRNAs have been found in the nuclear chromosomes with high sequence similarity. 150. In animals, the mitochondrial genome is typically a single circular chromosome that is approximately 16 kb long and has 37 genes. The genes, while highly conserved, may vary in location. Curiously, this pattern is not found in the human body. Laus, pediculus humanus. Instead, this mitochondrial genome is arranged in 18 minicircular chromosomes, each of which is 3-4 kb long and has 1 to 3 genes. 151. This pattern is also found in other sucking lice, but not in chewing lice. Recombination has been shown to occur between the minichromosomes. Human population genetic studies. Main article: Human mitochondrial genetics. The near absence of genetic recombination in mitochondrial DNA makes it a useful source of information for studying population genetics and evolutionary biology. 152. Because all the mitochondrial DNA is inherited as a single unit, or haplotype, the relationships between mitochondrial DNA from different individuals can be represented as a gene tree. Patterns in these gene trees can be used to infer the evolutionary history of populations. The classic example of this is in human evolutionary genetics, where the molecular clock can be used to provide a recent date for mitochondrial Eve. 153, 154. This is often interpreted as strong support for a recent modern human expansion out of Africa. 155. Another human example is the sequencing of mitochondrial DNA from Neanderthal bones. The relatively large evolutionary distance between the mitochondrial DNA sequences of Neanderthals and living humans has been interpreted as evidence for the lack of interbreeding between Neanderthals and modern humans. 156. However, mitochondrial DNA reflects only the history of the females in a population. This can be partially overcome by the use of paternal genetic sequences, such as the non-recombining region of the Y chromosome. 155. Recent measurements of the molecular clock for mitochondrial DNA, 157, reported a value of 1 mutation every 7,884 years. Dating back to the most recent common ancestor of humans and apes, which is consistent with estimates of mutation rates of autosomal DNA, 10-8 per base per generation. 158. Alternative genetic code. While slight variations on the standard genetic code had been predicted earlier, 159, none was discovered until 1979. When researchers studying human mitochondrial genes determined that they used an alternative code. 160. Nonetheless, the mitochondria of many other eukaryotes, including most plants, use the standard code. 161. Many slight variants have been discovered since 161, including various alternative mitochondrial codes. 162. Further, the AUA, AUC, and AUU codons are all allowable start codons. Some of these differences should be regarded as pseudo changes in the genetic code due to the phenomenon of RNA editing, which is common in mitochondria. In higher plants, it was thought that CGG encoded for tryptophan and not arginine. However, the codon in the processed RNA was discovered to be the UG codon, consistent with the standard genetic code for tryptophan. 163. Of note, the arthropod mitochondrial genetic code has undergone parallel evolution within a phylum, with some organisms uniquely translating AGG to lysine. 164. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Replication and inheritance. Main article, mitochondrial fission. Mitochondria divide by mitochondrial fission, a form of binary fission that is also done by bacteria. 165, although the process is tightly regulated by the host eukaryotic cell and involves communication between and contact with several other organelles. The regulation of this division differs between eukaryotes. In many single-celled eukaryotes, their growth and division are linked to the cell cycle. For example, a single mitochondrion may divide synchronously with the nucleus. This division and segregation process must be tightly controlled so that each daughter cell receives at least one mitochondrion. In other eukaryotes, in mammals for example, mitochondria may replicate their DNA and divide mainly in response to the energy needs of the cell, rather than in phase with the cell cycle. When the energy needs of a cell are high, mitochondria grow and divide. When energy use is low, mitochondria are destroyed or become inactive. In such examples, mitochondria are apparently randomly distributed to the daughter cells during the division of the cytoplasm. Mitochondrial dynamics. The balance between mitochondrial fusion and fission is an important factor in pathologies associated with several disease conditions. 166. The hypothesis of mitochondrial binary fission has relied on the visualization by fluorescence microscopy and conventional transmission electron microscopy, TEM. The resolution of fluorescence microscopy, almost equal to 200 nm, is insufficient to distinguish structural details, such as double mitochondrial membrane in mitochondrial division, or even to distinguish individual mitochondria when several are close together. Conventional TEM has also some technical limitations, which, in verifying mitochondrial division, cryo-electron tomography was recently used to visualize mitochondrial division in frozen hydrated intact cells. It revealed that mitochondria divide by budding. 167. An individual's mitochondrial genes are inherited only from the mother, with rare exceptions. 168. In humans, when an egg cell is fertilized by a sperm, the mitochondria, and therefore the mitochondrial DNA, usually come from the egg only. The sperm's mitochondria enter the egg, but do not contribute genetic information to the embryo. 169. Instead, paternal mitochondria are marked with ubiquitin to select them for later destruction inside the embryo. 170. The egg cell contains relatively few mitochondria, but these mitochondria divide to populate the cells of the adult organism. This mode is seen in most organisms, including the majority of animals. However, mitochondria in some species can sometimes be inherited paternally. This is the norm among certain coniferous plants, although not in pine trees and yews. 171. For mytilids, paternal inheritance only occurs within males of the species. 172, 173, 174, it has been suggested that it occurs at a very low level in humans. 175. Uniparental inheritance leads to little opportunity for genetic recombination between different lineages of mitochondria. Although a single mitochondrion can contain 210 copies of its DNA. 142. What recombination does take place maintains genetic integrity rather than maintaining diversity. However, there are studies showing evidence of recombination in mitochondrial DNA. It is clear that the enzymes necessary for recombination are present in mammalian cells. 176. Further, evidence suggests that animal mitochondria can undergo recombination. 177. The data are more controversial in humans, although indirect evidence of recombination exists. 178, 179. Entities undergoing uniparental inheritance and with little to no recombination may be expected to be subject to Muller's ratchet, the accumulation of deleterious mutations until functionality is lost. Animal populations of mitochondria avoid this build-up through a developmental process known as the Mount DNA bottleneck. The bottleneck exploits stochastic processes in the cell to increase the cell-to-cell variability in mutant load as an organism develops. A single egg cell with some proportion of mutant Mount DNA thus produces an embryo where different cells have different mutant loads. Cell-level selection may then act to remove those cells with more mutant Mount DNA, leading to a stabilization or reduction in mutant load between generations. The mechanism underlying the bottleneck is debated. 180, 181, 182, with a recent mathematical and experimental meta-study providing evidence for a combination of random partitioning of mTNAs at cell divisions and random turnover of Mount DNA molecules within the cell. 183. DNA repair. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Mitochondria can repair oxidative DNA damage by mechanisms analogous to those occurring in the cell nucleus. The proteins employed in Mount DNA repair are encoded by nuclear genes, and are translocated to the mitochondria. The DNA repair pathways in mammalian mitochondria include base excision repair, double-strand break repair, direct reversal and mismatch repair. 184, 185. Alternatively, DNA damage may be bypassed, rather than repaired, by translesion synthesis. Of the several DNA repair processes in mitochondria, the base excision repair pathway has been most comprehensively studied. 185. Base excision repair is carried out by a sequence of enzyme-catalyzed steps that include recognition and excision of a damaged DNA base, removal of the resulting abasic site, end processing, gap filling and ligation. A common damage in Mount DNA that is repaired by base excision repair is 8-oxyguanine produced by oxidation of guanine. 186. Double-strand breaks can be repaired by homologous recombinational repair in both mammalian Mount DNA, 187, and plant Mount DNA. 188. Double-strand breaks in Mount DNA can also be repaired by microhomology-mediated end joining. 189. Although there is evidence for the repair processes of direct reversal and mismatch repair in Mount DNA, these processes are not well characterized. 185. Lack of mitochondrial DNA. Some organisms have lost mitochondrial DNA altogether. In these cases, genes encoded by the mitochondrial DNA have been lost or transferred to the nucleus. 141. Cryptosporidium have mitochondria that lack any DNA, presumably because all their genes have been lost or transferred. 190. In Cryptosporidium, the mitochondria have an altered ATP generation system that renders the parasite resistant to many classical mitochondrial inhibitors such as cyanide, azide, and atovaquone. 190. Mitochondria that lack their own DNA have been found in a marine parasitic dinoflagellate from the genus Amoebophora. This microorganism, a kerati, has functional mitochondria that lack a genome. 191. In related species, the mitochondrial genome still has 3 genes, but in a kerati only a single mitochondrial gene, the cytochrome C oxidase gene, COX-1, is found, and it has migrated to the genome of the nucleus. 192. Dysfunction and disease. Mitochondrial diseases. Main article, Mitochondrial disease. Damage and subsequent dysfunction in mitochondria is an important factor in a range of human diseases due to their influence in cell metabolism. Mitochondrial disorders often present as neurological disorders, including autism. 18. They can also manifest as myopathy, diabetes, multiple endocrinopathy, and a variety of other systemic disorders. 193. Diseases caused by mutation in the MOUNT DNA include Kearns-Sayre syndrome, Meller's syndrome and Leber's hereditary optic neuropathy. 194. In the vast majority of cases, these diseases are transmitted by a female to her children, as the zygote derives its mitochondria and hence its MOUNT DNA from the ovum. Diseases such as Kearns-Sayre syndrome, Pearson syndrome, and progressive external ophthalmoplegia are thought to be due to large-scale MOUNT DNA rearrangements, whereas other diseases such as Meller's syndrome, Leber's hereditary optic neuropathy, MEF syndrome, and others are due to point mutations in MOUNT DNA. 193. It has also been reported that drug-tolerant cancer cells have an increased number and size of mitochondria which suggested an increase in mitochondrial biogenesis. 195. A 2022 study in Nature Nanotechnology has reported that cancer cells can hijack the mitochondria from immune cells via physical tunneling nanotubes. 196. In other diseases, defects in nuclear genes lead to dysfunction of mitochondrial proteins. This is the case in Friedreich's ataxia, hereditary spastic paraplegia, and Wilson's disease. 197. These diseases are inherited in a dominance relationship, as applies to most other genetic diseases. A variety of disorders can be caused by nuclear mutations of oxidative phosphorylation enzymes, such as coenzyme Q10 deficiency and Barth syndrome. 193. Environmental influences may interact with hereditary predispositions and cause mitochondrial disease. For example, there may be a link between explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF, pesticide exposure and the later onset of Parkinson's disease. 198, 199, other pathologies with etiology involving mitochondrial dysfunction include schizophrenia, bipolar disorder, dementia, Alzheimer's disease. 200. 201. Parkinson's disease, epilepsy, stroke, cardiovascular disease, myalgic encephalomyelitis / chronic fatigue syndrome, ME / CFS, retinitis pigmentosa, and diabetes mellitus. 202, 203. Mitochondria-mediated oxidative stress plays a role in cardiomyopathy in type 2 diabetics. Increased fatty acid delivery to the heart increases fatty acid uptake by cardiomyocytes, resulting in increased fatty acid oxidation in these cells. This process increases the reducing equivalents available to the electron transport chain of the mitochondria, ultimately increasing reactive oxygen species, ROS, production. ROS increases uncoupling proteins, UCPs, and potentiate proton leakage through the adenine nucleotide translocator, and the combination of which uncouples the mitochondria. Uncoupling then increases oxygen consumption by the mitochondria, compounding the increase in fatty acid oxidation. This creates a vicious cycle of uncoupling. Furthermore, even though oxygen consumption increases, ATP synthesis does not increase proportionally because the mitochondria are uncoupled. Less ATP availability ultimately results in an energy deficit presenting as reduced cardiac efficiency and contractile dysfunction. To compound the problem, impaired sarcoplasmic reticulum calcium release and reduced mitochondrial reuptake limits peak cytosolic levels of the important signaling ion during muscle contraction. Decreased intramitochondrial calcium concentration increases dehydrogenase activation and ATP synthesis. So in addition to lower ATP synthesis due to fatty acid oxidation, ATP synthesis is impaired by poor calcium signaling as well, causing cardiac problems for diabetics. 204. Mitochondria also modulate processes such as testicular somatic cell development, spermatogonal stem cell differentiation, luminal acidification, testosterone production in testes, and more. Thus, dysfunction of mitochondria in spermatozoa can be a cause for infertility. 205. In efforts to combat mitochondrial disease, mitochondrial replacement therapy, MRT, has been developed. This form of in vitro fertilization uses donor mitochondria, which avoids the transmission of diseases caused by mutations of mitochondrial DNA. 206. However, this therapy is still being researched and can introduce genetic modification, as well as safety concerns. These diseases are rare but can be extremely debilitating and progressive diseases, thus posing complex ethical questions for public policy. 207. Relationships to aging. See also, hallmarks of aging mitochondrial dysfunction. There may be some leakage of the electrons transferred in the respiratory chain to form reactive oxygen species. This was thought to result in significant oxidative stress in the mitochondria with high mutation rates of mitochondrial DNA. 208. Hypothesized links between aging and oxidative stress are not new and were proposed in 1956, 209, which was later refined into the mitochondrial free radical theory of aging. 210. A vicious cycle was thought to occur, as oxidative stress leads to mitochondrial DNA mutations, which can lead to enzymatic abnormalities and further oxidative stress. A number of changes can occur to mitochondria during the aging process. 211. Tissues from elderly humans show a decrease in enzymatic activity of the proteins of the respiratory chain. 212, however, mutated mount DNA can only be found in about 0.2% of very old cells. 213, large deletions in the mitochondrial genome have been hypothesized to lead to high levels of oxidative stress and neuronal death in Parkinson's disease. 214, mitochondrial dysfunction has also been shown to occur in amyotrophic lateral sclerosis. 215, 216. Since mitochondria cover a pivotal role in the ovarian function, by providing ATP necessary for the development from germinal vesicle to mature oocyte, a decreased mitochondria function can lead to inflammation, resulting in premature ovarian failure and accelerated ovarian aging. The resulting dysfunction is then reflected in quantitative, such as mount DNA copy number and mount DNA deletions, qualitative, such as mutations and strand breaks, and oxidative damage, such as dysfunctional mitochondria due to ROS, which are not only relevant in ovarian aging, but perturb oocyte cumulus crosstalk in the ovary, are linked to genetic disorders, such as Fragile X, and can interfere with embryo selection. 217. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. History. The first observations of intracellular structures that probably represented mitochondria were published in 1857, by the physiologist Albert von Kolliker. 218, 219, Richard Altman, in 1890, established them as cell organelles and called them bioblasts. 219, 220. In 1898, Carl Bender coined the term mitochondria from the Greek, mitos, thread, and chondrion, granule. 221, 219, 222. Leonia Michaelis discovered that Janus green can be used as a supravital stain for mitochondria in 1900. 223, in 1904, Friedrich Mivs made the first recorded observation of mitochondria in plants. In cells of the white water lily, Nymphea alba, 219, 224, and in 1908, along with Claudius Rigord, suggested that they contain proteins and lipids. Benjamin F. Kingsbury, in 1912, first related them with cell respiration, but almost exclusively based on morphological observations. 225, 219, in 1913, Otto Heinrich Warburg linked respiration to particles which he had obtained from extracts of guinea pig liver and which he called grana. 226, Warburg and Heinrich Otto Wieland, who had also postulated a similar particle mechanism, disagreed on the chemical nature of the respiration. It was not until 1925, when David Keeling discovered cytochromes, that the respiratory chain was described. 219. In 1939, experiments using minced muscle cells demonstrated that cellular respiration using one oxygen molecule can form 4-adenosine triphosphate, ATP, molecules, and in 1941, the concept of the phosphate bonds of ATP being a form of energy in cellular metabolism was developed by Fritz Albert Lippmann. In the following years, the mechanism behind cellular respiration was further elaborated, although its link to the mitochondria was not known. 219. The introduction of tissue fractionation by Albert Claude allowed mitochondria to be isolated from other cell fractions and biochemical analysis to be conducted on them alone. In 1946, he concluded that cytochrome oxidase and other enzymes responsible for the respiratory chain were isolated to the mitochondria. Eugene Kennedy and Albert Leoninger discovered in 1948 that mitochondria are the site of oxidative phosphorylation in eukaryotes. Over time, the fractionation method was further developed, improving the quality of the mitochondria isolated, and other elements of cell respiration were determined to occur in the mitochondria. 219. The first high-resolution electron micrographs appeared in 1952, replacing the Janus green stains as the preferred way to visualize mitochondria. 219. This led to a more detailed analysis of the structure of the mitochondria, including confirmation that they were surrounded by a membrane. It also showed a second membrane inside the mitochondria that folded up in ridges dividing up the inner chamber, and that the size and shape of the mitochondria varied from cell to cell. The popular term powerhouse of the cell was coined by Philip Sienkiewicz in 1957. 4. 227. In 1967, it was discovered that mitochondria contained ribosomes. 228. In 1968, methods were developed for mapping the mitochondrial genes, with the genetic and physical map of yeast mitochondrial DNA completed in 1976. 219. See also antimitochondrial antibodies, mitochondrial metabolic rates, mitochondrial permeability transition pore, mitophagy, Nebenkern, oncocyte, oncocytoma, paternal mount DNA transmission, plastide, submitochondrial particle. References: 1 Mitochondrion, Lexico UK English Dictionary, Oxford University Press. Archived from the original on 2 January 2020. 2 Campbell Na, Williamson B., Hayden R.J., 2006. Biology, Exploring Life. Boston, Massachusetts, Pearson/Prentice Hall. ISBN 978-0132508827. Archived from the original on 2 November 2014. Retrieved 6 January 2009. 3 Mighty Mitochondria and Neurodegenerative Diseases. Science in the News. 1 February 2012. Archived from the original on 6 April 2022. Retrieved 24 April 2022. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 4 A.B. Sienkiewicz P. 1957. Powerhouse of the Cell. Scientific American. 197, 1131-140. Bibcode, 1957 SCIM.197A.131S. Do, 10.1038 / American 0757-131. 5 ABCD Kankowska, Varsik V, Zuboff Z, Triatli SC, Petrilkov R, Imel, it al. May 2016. A eukaryote without a mitochondrial organelle. Current Biology. 26, 10 1274 1284. Bibcode, 2016 CBio, 26.1274K. Do, 10.1016/j.cub.2016.03.053. PMID 27185558. 6 LePage M. Animal that doesn't need oxygen to survive discovered New Scientist. New Scientist. Archived from the original on the 26th of February 2020. Retrieved the 25th of February 2020. 7 AB Yahalom ED, Atkinson SD, Newhoff M, Chang S, Philippe H, Cartwright P, et al. March 2020. A cnidarian parasite of salmon, Mixozo, Hennegaia, lacks a mitochondrial genome. Proceedings of the National Academy of Sciences of the United States of America. 117, 10 5358 5363. Bib code 2020 NAS 117.5358 Do 10.1073//NAS.19099071117. PMC 7071853. PMID 32094163. 8 AB Hens K, Martin W, November 2003. Evolutionary Biology, Essence of Mitochondria. Nature. 426, 6963 127128. Bib code 2003 Nature.426127H. Do 10.1038//4261227A. PMID 146144484. S2 SID 862398. 9 Ledger mm, Colisco M, Kamikawa R, Stairs CW, Coombe K, Epica I, et al. April 2017. Organelles that illuminate the origins of Trichomonas hydrogenosomes and chidiomitosomes. Nature, Ecology and Evolution. 1, 40092. Bib code 2017 NATE 1, 92L. Do 10.1038//41559-017-0092. PMC 5411260. PMID 28474007. 10 AB Novak LV, Triatli SC, Piri J, Halakak P, Pi Palyar SV, Varsik V, et al. December 2023. Dutcher SK, ed. Genomics of Prexostyler flagellates illuminates the path towards the loss of mitochondria. PLOS Genetics. 19, 12E10111050. Do 10.1371//journal.pgen.10111050. PMC 107032772. PMID 38060519. 11 Wemer/L, Lee D, March 2016. Quantification of mitochondrial morphology in neurites of dopaminergic neurons using multiple parameters. Journal of Neuroscience Methods. 262, 5665. Do 10.1016/j.newmeth.2016.01.008. PMC 4775301. PMID 267777473. 12 ABC McBride HM, Naspil M, Wajik S, July 2006. Mitochondria, more than just a powerhouse. Current Biology. 16, 14. R551 R560. Bibcode 2006 CBio 16.R551M. Do 10.1016/j.cub.2006.06.054. PMID 16860735. S2 SID 16252290. 13 Carat Valero T, 2014. Mitochondrial biogenesis, pharmacological approaches. Current pharmaceutical design. 20, 35 5507 5509. Do 10.200116/13816/128120351409111421118. HDL 10454/13341. PMID 246069795. Mitochondrial biogenesis is therefore defined as the process via which cells increase their individual mitochondrial mass. 3. Mitochondrial biogenesis occurs by growth and division of pre-existing organelles and is temporally coordinated with cell cycle events. 1. 14 Sanchez Gamar F. Garcia Jimenez J.L., Gomez Cabrera M.C., Palado F.V., 2014. Mitochondrial biogenesis in health and disease. Molecular and therapeutic approaches. Current pharmaceutical design. 20, 35 5619 5633. Do 10.2001/13816128661403060951006. PMID 246060801. Mitochondrial biogenesis, MB, is the essential mechanism by which cells control the number of mitochondria. 15 Gardner, Bowles R.G., 2005. Is a mitochondrial psychiatry in the future? A review. Car. Psychiatry Revelation 1, 32557271. Do 10.200174/15734000577457506. 16 Lesnovsky E.J., Mogadas S., Tandala B., Kerner J., Hoppel C.L., June 2001. Mitochondrial dysfunction in cardiac disease. Ischemia, reperfusion, aging, and heart failure. Journal of Molecular and Cellular Cardiology. 33, 6 1065 1089. Do 10.1006/JMCC.2001.1378. PMID 11444914. 17 Dawn G.W., Vega R.B., Kelly D.P., October 2015. Mitochondrial biogenesis and dynamics in the developing and diseased heart. Genes and development. 29, 1981 1991 do 10.1001/GAD.2698994.115. PMC 46043339. PMID 264438444. 18 A.B. Griffiths K.K., Levi R.J., 2017. Evidence of mitochondrial dysfunction in autism, biochemical links, genetic-based associations, and non-energy related mechanisms. Oxidative medicine and cellular longevity. 2017, 43140025. Do 10.1155/2017/43144025. PMC 5467355. PMID 2863065. 19 carat Nepal, May 2011. Normal and disordered reticulocyte maturation. Current opinion in hematology. 18.3152157. Do. 10.1097/moh.0b013E328345213E. PMC 3157046. PMID 214230015. 20 carat ABCDEFGHIJ Alberts B, Johnson O, Lewis J, Raff M, Roberts K, Walter P, 2005. Molecular Biology of the Cell. New York. Garland Publishing Incorporated. ISBN 978-0815341055. 21 carat ABCDEFGHIJKL Voet D, Voet JC, Pratt CW, 2006. Fundamentals of Biochemistry, 2nd ed. John Wiley & Sons, Incorporated. pp. 547, 556. ISBN 978-0471214953. 22 carat Anderson S.G., Karlbergo, Canbach B., Kurland C.G., January 2003. On the Origin of Mitochondria, A Genomics Perspective. Philosophical Transactions of the Royal Society of London. Series B, Biological Sciences. 358, 1429 16577, discussion 1779. Do. 10.1098/rstb.2002.1193. PMC 1693097. PMID 125949925. 23 carat AB Gabalden T, October 2021. Origin and Early Evolution of the Eukaryotic Cell. Annual Review of Microbiology. 75, 1631. 647 do. 10.1146/An of Micro 090817-062213. PMID 343430017. S2 SID 236916203. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 24 carat Mitochondrion, much more than an energy converter. British Society for Cell Biology. Archived from the original on April 4, 2019. Retrieved the 19th of August 2013. 25 carat Blackledison E, Fort AM, September 2001. VDAC channels. EUBM Life. 52, 35113118. Do. 10.1080/15216540152845902. PMID 11798022. S2 SID 38314888. 26 carat Hugenboom BW, Suda K, Engler, Fauchardis D, July 2007. The supramolecular assemblies of voltage-dependent ion channels in the native membrane. Journal of Molecular Biology. 370, 22462552. do. 10.1016/J.JMB.2007.04.073. PMID 175244423. 27 ZK, June 2010. Structure and evolution of mitochondrial outer membrane proteins of beta-barrel topology. Biochimica. IT Biophysica Acta, BBA, Bioenergetics. 1797, 67 1292 1299 do. 10.1016/j.baybio.2010.04.019. PMID 204508883. 28 AB Herman J.M., Newport W., April 2000. Protein Transport into Mitochondria, PDF. Current Opinion in Microbiology. 3, 2. 210214. Do. 10.1016/1369-5274, 000077-1. PMID 1074497. Archived, PDF, from the original on 16 August 2022. Retrieved 10 July 2022. 29 AB Chipuk J.E., Buchia Hayes L., Green Dr., August 2006. Mitochondrial outer membrane permeabilization during apoptosis. The innocent bystander scenario. Cell death and differentiation. 13. 8 1396 1402 do. 10.1038/sj.cdd.44019663. PMID 167103662. 30 ABCDE Hayashiti, Rizuto R., Hajnakskiji, Su TP, February 2009. Ma'am, more than just a housekeeper. Trends in Cell Biology. 19. 28188 do. 10.1016/j.tcb.2008.12.002. PMC 2750097. PMID 191445519. 31 Skenkel LC, Bakovic M., January 2014. Formation and regulation of mitochondrial membranes. International Journal of Cell Biology. 2014, 709828. Do. 10.101155/2014/7098828. PMC 3918842. PMID 245787808. 32 Macmillan J.B., Dowhan W., December 2002. Cardiolipin and apoptosis. Biochimica it Biophysica Acta, BBA, Molecular and Cell Biology of Lipids. 1585, 2397-107 do. 10.1016/s1388-1981, 02-00329-3. PMID 125315542. 33 Bautista J.S., Falabella M., Flannery P.J., Hannah M.G., Heels S.J., Pope Sat. It A.L. December 2022. Advances in methods to analyze cardiolipin and their clinical applications. Trends in analytical chemistry. 157, 116808. Do. 10.1016/j.track.2022.116808. PMC 7614147. PMID 367515553. S2 SID 253211400. 34 Ule R.J. van der Bleekem, August 2012. Mitochondrial fission, fusion, and stress. Science. 337, 6098 1062 1065. Bibcode, 2012 SCI, 337.1062Y. Do, 10.1126/science.1219855. PMC 4762028. PMID 2293670. 35 Xarep C, POS 5B, Schwartz AD, Deans A, 2018. Mitochondrial ultrastructure is coupled to synaptic performance at axonal release sites. eNeuro 5, 1N0.039017.2018 do, 10.1523/N0.0390-17.2018. PMC 5788698. PMID 2938328. 36 Manila CA, 2006. Structure and dynamics of the mitochondrial inner membrane crystal. Biochimica et Biophysica Acta. BBA, Molecular Cell Research. 1763, 56542548 10.1016/j.bamca.2006.04.006. PMID 167308111. 37 Bogenhagen DF, September 2012. Mitochondrial DNA nuclear structure. Biochimica et Biophysica Acta, BBA, Gene. Regulatory mechanisms. 1819, 910914922, 10.1016/j.bagm.2011.11.005. PMID 22142616. 38 Rich PR, December 2003. The molecular machinery of Keelin's respiratory chain. Biochemical Society Transactions. 31, pt 6. 1095 1105. Do, 10.1042/BST03111095. PMID 146410005. 39 Stoimenova M, Agamadi Evor, Gupta KJ, Hill Road, July 2007. Nitrite-driven anaerobic ATP synthesis in barley and rice root. Mitochondria. Planter. 226, 2465474. Bibcode, 2007 plant.226465s. Do, 10.1074/S00425-007-0496-0. PMID 1733352. S2 SID 896350. 40 Newport W, 1997. Protein import into mitochondria. Annual Review of Biochemistry. 66, 863917. Do, 10.101146/anav.biochem.66.1.863. PMID 92422927. 41 ABCDEF Stria L, 1995. Citric acid cycle. In Biochemistry, 4th ed. New York, W.H. Freeman and Company. pp. 509. 527, 569579, 614616, 638641, 73275, 739748, 77773. ISBN 0716720094. 42 Kingo, Salakma, Gottlieb, August 2006. Succinate dehydrogenase and fumarate hydratase, linking mitochondrial dysfunction and cancer. Oncogen. 25, 34 4675 4682 do. 10.1038/sj.onc.12095994. PMID 16892081. S2 SID 26263513. 43 AB Voet D, Voet JG, 2004. Biochemistry, 3rd ed. New York, NY, Wiley. P 804. ISBN 978-0-471-19350-0. 44 AB Atkins P. De Paula J. 2006. Impact on Biochemistry, Energy Conversion in Biological Cells. Physical Chemistry, 8th ed. New York, Freeman. pp. 225229. ISBN 978-0-7167-8759-4. 45 Huang H. Manton kg, May 2004. The Role of Oxidative Damage in Mitochondria During Aging, a Review, PDF. Frontiers in Bioscience. 9, 131117 do. 10.2741/1298. PMID 149775532. S2 SID 22782119. Archived from the original, PDF. On 3 March 2019. 46 Mitchell P., Moyle J., January 1967. Chemiosmotic Hypothesis of Oxidative Phosphorylation. Nature. 213, 5072 137139. Bibcode, 1967 Nature.213137m. Do. 10.1038/213137/0. PMID 4291593. S2 SID 41499605. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 47 Mitchell P., June 1967. Proton current flow in mitochondrial systems. Nature. 214, 5095 1327 1328. Bibcode, 1967 Nature.214.1327m. Do. 10.1038/21413270. PMID 6056845. S2 SID 41600146. 48 Nobel Foundation. Chemistry 1997. Archived from the original on 8 July 2007. Retrieved 16 December 2007. 49 AB Mozo J, M Ray Y, Builord F, Rukia D, Criskilo F, November 2005. Thermoregulation, what role for UCPs in mammals and birds? Bioscience Reports 25, 34227249 do 10.1007-407-005-2887-4 PMID 16283555 S2 SID 164450 50 carat Castaniotis Orshio KJ, Keratar JM, Montua SG, Maklam, Nera it al. January 2017. Mitochondrial fatty acid. Synthesis, fatty acids and mitochondrial physiology. Biochimica et Biophysica Acta, BBA, Molecular and Cell Biology of Lipids. 1862, 139-48 do. 10.1016.4/j.bilip.2016.08.011 PMID 2755347 51 carat Clay HB, Parle AK, Mitchell SL, Singh L, Bell LN, Murdoch DG, March 2016. Peterson J, ed. Altering the mitochondrial fatty acid synthesis, Mount FASII, pathway modulates cellular metabolic states and bioactive lipid profiles as revealed by metabolomic profiling. PLOS 1. 11, 3E01511171. Bib code, 2016 PLOSO 11511171C. Do. 10.1371/journal.pone.0151171. PMC 47862887. PMID 26963735. 52 carat ABC Nowinski SM, Van Vranken JG, Dove KK, Rutter J, October 2018. Impact of mitochondrial fatty acid synthesis on mitochondrial biogenesis. Current biology. 28, 20 R1212 R1219. Bib code, 2018 C-Bio, 28 R1212N. Do. 10.1016/j.cub.2018.08.022. PMC 6258005. PMID 30352195. 53 carat Web Z, Beringer S, Alata BK, Watkins D, Rosenblatt D, Spiker Kota U, it al. November 2019. The emerging role of the mitochondrial fatty acid synthase, Mount FASII, in the regulation of energy metabolism. Biochimica et Biophysica Acta, BBA. Molecular and Cell Biology of Lipids. 1864, 11 1629 1643 do. 10.1016/j.bilip.2019.07.012. PMID 31376476. S2 SID 1994049006. 54 carat Santuli G, Xi W, Recon SR, Marks R, September 2015. Mitochondrial calcium overload is a key determinant in heart failure. Proceedings of the National Academy of Sciences of the United States of America. 112, 36 1138911394. Bib code, 2015 NAS 11211389S. Do. 10.1073/NAS.1513047112. PMC 456867. PMID 26217001. 55 AB Ziegel GJ, Agranov BW, Fisher SK, Albus RW, Uller MD, eds. 1999. Basic Neurochemistry, 6 ed. Lippincott Williams and Wilkins. ISBN 978-0397518203. 56 AB Rossier MF, August 2006. T-channels and steroid biosynthesis, in search of a link with mitochondria. Cell Calcium. 40, 2. 155164. Do. 10.1000.J.Seca.2006.04.020. PMID 167599697. 57 Brighton CT, Hunt RM, May 1974. Mitochondrial calcium and its role in calcification. Histochemical localization of calcium in electron micrographs of the epiphyseal growth plate with K-pyrone T-monate. Clinical Orthopedics and Related Research. 100. 54064162. 10.1000.000030860197403005000000057. PMID 413494. 58 Brighton CT, Hunt RM, July 1978. The role of mitochondria in growth plate calcification as demonstrated in a rachitic model. The Journal of Bone and Joint Surgery. American Volume 60, 563639 10.2001664623-197860050-00007. PMID 681381. 59 ABC Santuliji, Marks R. 2015. Essential roles of intracellular calcium release channels in muscle, brain, metabolism, and aging. Current molecular pharmacology. 8, 22062222. 10.2001-1874467208661505071051005. PMID 2596694. 60 Pitso P. Potson T. October 2007. Mitochondria endoplasmic reticulum choreography, structure and signaling dynamics. Trends in cell biology. 17, 10.511517 do. 10.1016/J.TCB.2007.07.011. PMID 1785178. 61 AB Miller R.J., March 1998. Mitochondria, the Kraken Wakes. Trends in Neurosciences. 21, 39597 do. 10.1016/S0166. 2236, 97, 01206X. PMID 95309113. S2 SID 519381. 62 Santuliji, Paganoji, Sadhu C, Xi W, Recon S, Diasia SL, IT AL. May 2015. Calcium release channel RYR2 regulates insulin release and glucose homeostasis. The Journal of Clinical Investigation. 125, 5 1968-1978 do. 10.1172/JCI 79273. PMC 44632204. PMID 258448899. 63 carat Schwarlander M, Logan DC, Johnston IG, Jones NS, Mayer H, Fricker MD, et al. March 2012. Pulsing of membrane. Potential in individual mitochondria, a stress-induced mechanism to regulate respiratory bioenergetics in Arabidopsis. The plant cell. 24, 3 1188 1201 do. 10.1105/TPC.112.0964338. PMC 3336130. PMID 223954886. 64 carat Ivanikov MV, McLeod GT, June 2013. Mitochondrial free Ca² levels and their effects on energy metabolism in Drosophila. Motor nerve terminals. Biophysical Journal. 104, 11 2353 2361. Bib code, 2013 BPJ. 104.2353 do. 10.1016/J.BPJ.2013.03.064. PMC 3672877. PMID 237460507. 65 carat Weinberg F, Schandl NS, October 2009. Mitochondrial Metabolism and Cancer. Annals of the New York Academy of Sciences. 1177, 16673. Bib code, 2009 NYSA 1177, 66W. Do. 10.1146J.1049/6632.2009.05039.x. PMID 198456008. S2 SID 2982752. 66 carat AB Moreno Sanchez R, Rodriguez Enriquez S, Marin Hernandez A, Saavedra E, March 2007. Energy Metabolism in Tumor Cells. The Febs Journal. 274, 6 1393 1418 do. 10.1142/J.1758.2007.05686.x. PMID 1730274. S2 SID 77481115. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 67 carat Mystery JJ, Marline CR, Moore JA, Helmick C, Wojtowicz EE, Smith JG, et al. December 2019. ROS-mediated Pi3K activation drives mitochondrial transfer from stromal cells to hematopoietic stem cells in response to infection. Proceedings of the National Academy of Sciences of the United States of America. 116, 492461024619. Bib code, 2019 NAS 116244610M. Do. 10.1073/NAS.19132781116. PMC 69000710. PMID 31727843. 68 carat Peterson PL, December 1994. ATP synthase. the machine that makes ATP. Current biology. 4. 12 1138 1141. Bibcode 1994 CBio 4.1138p. Do 10.1016/S0960-9822 00000257-8. PMID 77045882. S2 SID 10279742. 69 Patapaji, Hayward HK, De Bruijn JD, Leather, October 2011. The metabolism of human mesenchymal stem cells during proliferation and differentiation. Journal of Cellular Physiology. 226, 10 2562 2572 10.1002/JCP.22605. PMID 21792913. S2 SID 222598833. 70 Agarwal B, June 2011. A role for onions in ATP synthesis and its molecular mechanistic interpretation. Journal of Bioenergetics and Biomembranes. 43, 3299310 10.1007/S1086-3-011-9358-3. PMID 21647635. S2 SID 297153883. 71 ABC Suite S, Singji, July 1999. Changes in mitochondrial mass, membrane potential, and cellular adenosine triphosphate content during the cell cycle of human leucemic, HL60, cells. Journal of Cellular Physiology. 180, 190-196. Do 10.1002/CSI 1097-4652, 199907, 181.3.0.co, 2-6. PMID 10362021. S2 SID 25906-279. 72 Ria Romo M, August 2021. Cell death as part of innate immunity, cause or consequence. Immunology. 163, 4399. 415 Do 10.1111/im.13325. PMC 82741779. PMID 33682112. 73 Green Dr. September 1998. Apoptotic Pathways, The Roads to Ruin. Cell. 94, 6695698 Do 10.1016/S0092. 8674, 00, 81728-6. PMID 9753316. S2 SID 166548082. 74 ABCDEF Bahato, Macvicar T, Langer T, the 27th of July 2021. Metabolism and innate immunity meet at the mitochondria. Frontiers in Cell and Developmental Biology. 9, 720490. Do 10.3389/FSL.2021.720490. PMC 835326. PMID 34386501. 75 ABCDEF Murphy MP, Anil LA, February 2024. A break in mitochondrial endosymbiosis as a basis for inflammatory diseases. Nature. 626, 7998 271279. Bibcode 2024 nature.626271m. Do 10.1034/S41586-023-06866Z. PMID 383265990. 76 carat Crisco DV, Agostiniz P, Crisco O, Gargay D, Bashert C, Lambrecht BN, it al. April 2011. Emerging role of damage-associated molecular patterns derived from mitochondria in inflammation. Trends in immunology. 32, 415764. Do 10.1016/J.it.2011.01.005. PMID 21334975. Cited in 75. 77 carat Riley J.S., Tate S.W., April 2020. Mitochondrial DNA in inflammation and immunity. EMBO reports. 21, 4E49799. Do 10.1524/emb.2019497999. PMC 71322203. PMID 3220265. Cited in 75. 78 carat Seth R.B., Sun L., EACK, Chen Z.J., September 2005. Identification and characterization of MAVS, a mitochondrial antiviral signaling protein that activates NF-kappa and IRF3. Cell 122, 5669682 do 10.1016/J.cell.2005.08.012. PMID 161257663. Cited in 75. 79 carat Dorwood The, Lucas C.D., Doherty M.K., Chapman G.B., Scalifield E.J., Conway Morris O., it al. October 2017. Novel role for endogenous mitochondrial formylated peptide-driven formal peptide receptor 1 signaling in acute respiratory distress syndrome. Thorax. 72, 10928936 do 10.1136/thoraxgen 2017-210030. PMC 5738532. PMID 2846931. Cited in 75. 80 carat CAIN, Gomez Durano, Yonova Duini, Kundu K, Burgess AI, Golder ZJ, it al. September 2021. Mitochondrial DNA variants modulate N-formylmethionine, proteostasis and risk of late-onset human diseases. Nature Medicine. 27, 9 1564 1575. Do 10.1038/S41591-021-01441-3. PMID 344267006. Cited in 75. 81 carat Zhang W, Wang G, Zhu ZG, 2H, Hu F, Dai J, it al. June 2019. Lactate is a natural suppressor of RLR signaling by targeting MAVS. Cell 178, 1176189.E15 do 10.1016/j.cell.2019.05.003 PMC 66253551 PMID 311552331. Cited in 74. 82 Porcelot M. Arnold D. September 2014. Mitochondrial Dynamics and the Innate Antiviral Immune Response. The Febs. Journal. 281, 17 3791 3802 do. 10.1111/Febs.12940. PMID 250519991. Cited in 74. 83 Li X, Fang P, Mai J, Choi E T, Wang H, Yang XF, February 2013. Targeting mitochondrial reactive oxygen species as novel therapy for inflammatory diseases and cancers. Journal of Hematology and Oncology. 6, 1919 do. 10.1156/1756-8722-6-19. PMC 3599349. PMID 234428817. 84 Hajnowski G, Xorders G, Das S, Garcia Perez C, Sao Tome M, Sinaroy S, et al. 2006. Mitochondrial calcium signaling and cell death, approaches for assessing the role of mitochondrial Ca2+ uptake in apoptosis. Cell calcium. 40, 56553560. Do. 10.1016/j.seca.2006.08.016. PMC 2692319. PMID 17074387. 85 Ohama T, August 1997. Evolutionary Consideration on 5-aminolevulonate synthase in Nature. Origins of Life and Evolution of the Biosphere. 27, 44054112. Bibcode 1997 OLEB 274050. Do. 10.1023/100658360141. PMID 9249985. S2 SID 136028777. 86 Clinch cm, December 2008. Estrogenic Control of Mitochondrial Function and Biogenesis. Journal of Cellular Biochemistry. 105, 6 1342 1351 do. 10.1002/JCB.21936. PMC 2593138. PMID 188465005. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 87 Alvarez Delgado C, Mendoza Rodriguez C.A., Picasso O, Serban M, August 2010. Different expression of alpha and beta. Mitochondrial estrogen receptors in the aging rat brain, interaction with respiratory complex V. Experimental Gerontology. 45, 7. 858585. Do. 10.1016/j.exja.2010.01.015. PMID 200967665. S2 SID 308411790. 88 Pavon N, Martinez Abundis E, Hernandez L, Gallardo Perez J.C., Alvarez Delgado C., Serban M, et al. October 2012. Sexual hormones, effects on cardiac and mitochondrial activity after ischemia reperfusion in adult rats. Gender difference. The Journal of Steroid Biochemistry and Molecular Biology. 132, 12135146 10.1016/J.JSBMB.2012.05.003. PMID 226093114. S2 SID 247944040. 89 Breeder CN, Devanzo GG, Barso PJ, Sarava Cameron O, Maurice Vieira PM, September 2019. Mitochondria as central hub of the immune system. Redox Biology. 26, 101255. Do 10.1016/J.redox.2019.101255. PMC 6598836. PMID 312475505. 90 AB Xarep C, Pos 5B, Leonard N, Ficata, Lazalo ZI, Leal Z, it al. January 2020. Microglia monitor and protect neuronal function through specialized somatic purinergic junctions. Science. 367, 6477 5285537. Bibcode 2020 SCI 3675228C. Do 10.1126/science.arc6752. PMID 318311638. S2 SID 209343260. Archived from the original on the 2nd of May 2023. Retrieved the 10th of February 2022. 91 Xarep C, Schwartz AD, Pos 5B, Lazalo ZI, Keleamea, Kanaya Z, it al. September 2022. Microglial control of neuronal development via somatic purinergic junctions. Cell reports. 40, 12 111369 do 10.1016/J.cellrip.2022.111369. PMC 9513806. PMID 361304888. S2 SID 252416407. 92 Taylor SW, Fahii, Chang B, Glenn GM, Warnock Do, Wiley S, it al. March 2003. Characterization of the human heart. Mitochondrial proteome. Nature Biotechnology. 21, 32812886 do 10.1038/NBT793. PMID 125924111. S2 SID 2732951. 93 Zhang J, Li X, Mueller M, Wang Y, Zong C, Deng N, it al. April 2008. Systematic characterization of the murine mitochondrial proteome using functionally validated cardiac mitochondria. Proteomics. 8, 1564 1575 do 10.1002/PMIC.2007000851. PMC 2799225. PMID 183483319. 94 Zhang J, Lai M. The, Mueller M, Wang Y, Zong C, Deng N, et al. June 2008. Altered proteome biology of cardiac mitochondria. Under stress conditions. Journal of Proteome Research. 7, 6 2204 2214 do. 10.1021/PR070371F. PMC 3805274. PMID 184844766. 95 Logan DC, June 2010. Mitochondrial fusion, division and positioning in plants. Biochemical Society Transactions. 38, 3789. 795 do. 10.1042/BST03807889. PMID 204916666. 96 Das Neves RP, Jones NS, Andrew L., Gupta R., Enver T., Ibora FJ, December 2010. Weissman JS, ed. Connecting Variability in Global Transcription Rate to Mitochondrial Variability. PLOS Biology. 8, 12E10000560 do. 10.1300.journal.bio.10000560. PMC 3001896. PMID 211799497. 97 Johnston IG, Guile B., Neves RP, Enver T., Ibora FJ, Jones NS, 2012. Hoare JM, ed. Mitochondrial variability as a source of extrinsic cellular noise. PLOS Computational Biology. 8, 3E10024116R14, 1107.4499. Bibcode, 2012 PLSCB, 8E2416J. Do. 10.1371/journal.pb.10022416. PMC 3297557. PMID 2241233. 98 Rappaport L., Oliviero P., Samuel J. L., July 1998. Cytoskeleton and mitochondrial morphology and function. Molecular and cellular biochemistry. 184, 12101105 do. 10.1023/100684311316. PMID 9746315. S2 SID 28165195. 99 Hoyt-Singh H., Johnston IG, Jones NS, June 2015. What is the function of mitochondrial networks? A theoretical assessment of hypotheses and proposal for future research. Bioessays. 37, 6687700 do. 10.1002/bis.2014000188. PMC 4672710. PMID 2584785. 100 Saltis BJ, Gupta RS, 1992. Interrelationships of endoplasmic reticulum, mitochondria, intermediate filaments, and microtubules. A quadruple fluorescence labeling study. Biochemistry and cell biology equals biochimie et biologie cellulaire. 70, 10. 11 1174 1186. Do. 10.1139/092-163. PMID 1363623. 101 Tang HL, Lung HL, Wu KC, La, Tang HM, Fung MC, February 2008. Vimentin supports mitochondrial morphology and organization. The Biochemical Journal. 410, 11411462, 10.1042/BJ20071072. PMID 17983357. 102 ABCDEFGHIJKLMN Rizzuto R, March ES, Bonera M, Augieri P, Bonania, De Stefani D, et al. November 2009. CA 2+. Transfer from the to mitochondria, when, how and why. Biochimica et Biophysica Acta, BBA, Bioenergetics. 1787, 11. 1342 1351. Do. 10.1016/j.bay.bio.2009.03.015. PMC 27304223. PMID 19341702. 103 AB de Brito OM, Scarano L, August 2010. An intimate liaison, spatial organization of the endoplasmic reticulum mitochondria. Relationship. The EMBO Journal. 29, 16 2715 2723 do. 10.1038/embodge.2010.177. PMC 29246551. PMID 2071741. 104 AB Vance J. Xiao YJ, 1996. Intracellular trafficking of phospholipids, import of phosphatidylserine into mitochondria. Anticancer Research. 16, 3B 1333 1339. PMID 86944499. 105 ABC Lebedzinska M, Sabaidkai G, Jones A.W., Duzinski J, Wikowski Mr., October 2009. Interactions between the endoplasmic reticulum, mitochondria, plasma membrane and other subcellular organelles. The International Journal of Biochemistry and Cell Biology. 41, 10/18/05/1816 do. 10.1016/j.biosol.2009.02.017. PMID 19703651. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 106 Twig G, Elorza, Molina H, Mohammed H, Wikstrom J.D., Waza G, et al. January 2008. Fission and selective fusion govern mitochondrial segregation and elimination by autophagy. The EMBO Journal. 27, 24334446. Do. 10.1038/sj.embodge.7601963. PMC 223439. PMID 18200046. 107 ABCDEF Osman C, Voelka Dr, Langer T, January 2011. Making heads or tails of phospholipids in mitochondria. The Journal of Cell Biology. 192, 1716 do. 10.1083/jcb.2010061559. PMC 3019561. PMID 212200505. 108 Kahneman B, Curry E, Collins S R, Schuldner M, Nunnery J, Weissman J S, et al. July 2009. Inner mitochondria tethering complex revealed by a synthetic biology screen. Science 325, 5939 477481. Bibcode 2009 SCI 325477K. Do 10.1126/science.1175088. PMC 2933203. PMID 195564661. 109 Rusenol A, Qui Z, Chen MH, Vance J.E., November 1994. A unique mitochondria-associated membrane fraction from rat liver. Has a high capacity for lipid synthesis and contains pre-Golgi secretory proteins including nascent lipoproteins. The Journal of Biological Chemistry 269, 442749427502 do 10.1016/S0021-9258, 18, 47012-3. PMID 7961664. 110 AB Kopako, Kruglikov I, Pivnova-T, Voitenko N, Fedorko N, May 2008. Functional coupling between ryanodin receptors. Mitochondria and Ca2+, ATPase in rat submandibular acinar cells. Cell calcium 43, 5469481. Do 10.1016/J.Seca.2007.08.001. PMID 1788947. 111 Ksorders G, Hajnowski G, April 2001. Sorting of calcium signals at the junctions of endoplasmic reticulum and mitochondria. Cell calcium 29, 4249262 do 10.1054/Seca.2000.0191. PMID 1124393. 112 ABCD Decuperia JP, Monaco G, Bultink G, Missian L, De Smeet H, Paris JB, May 2011. The IP 3 receptor mitochondria. Connection in apoptosis and autophagy. Biochimica et Biophysica Acta, BBA, Molecular Cell Research. 1813, 51003 1013. Do 10.1016/J.Bamka.2010.11.023. PMID 211465662. 113 Diox BP, Flygert R, Gus R, June 2017. MagFluO4 in T cells, imaging of intra-organelle-free Ca2+ concentrations. Biochimica et Biophysica Acta, BBA, Molecular Cell Research. 1864, 6977986 do 10.1016/J.Bamka.2016.11.026. PMID 279132206. 114 Hajnowski G, Ksorders G, YM, 2011. Old Players in a New Role, Mitochondria-Associated Membranes, VDAC, and Ryanodin. Receptors as contributors to calcium signal propagation from endoplasmic reticulum to the mitochondria. Cell calcium 32, 56. 363377. Do 10.1016/S0143416002001872. PMID 125430996. 115 Marriott KS, Prasadem, Thaplil V, Bose HS, December 2012. 1 receptor at the mitochondrial-associated endoplasmic reticulum membrane is responsible for mitochondrial metabolic regulation. The Journal of Pharmacology and Experimental Therapeutics. 343, 3578586 10.1124/chbit.112.19816.8 PMC 35000540 PMID 229233735 116 Hayashiti, Su TP, November 2007. Sigma 1 receptor chaperones at the mitochondrion interface regulate Ca2+ signaling and cell survival. Cell 131, 3596610 10.1010.j.cell.2007.08.036 PMID 179811125 S2 SID 1888568 117 Ksorders G, Weaver D, Hajnowski G, July 2018. Endoplasmic reticulum mitochondrial contactology, structure and signaling. Functions. Trends in cell biology. 28, 7523542. 10.1016/j.tcb.2018.02.009 PMC 6005738 PMID 295888129 118 McCutcheon JP, October 2021. The Genomics and Cell Biology of Host Beneficial Intracellular Infections. Annual Review of Cell and Developmental Biology. 37, 111542 10.1146/An of Cell Bio 120219-024122. PMID 34242059. S2 SID 235786110. 119 Kalia V, the 8th of June 2022. Mitochondria and the Origin of Eukaryotes. Knowable Magazine. Do 10.1146/knowable 0608222. 2. S2 SID 249526889. Archived from the original on 15 August 2022. Retrieved 18 August 2022. 120 AB Margulies L, Sargon D, 1986. Origins of Sex, 3 billion years of genetic recombination. New Haven, CT, Yale University Press. pp. 6971, 87. ISBN 978-0300033403. 121 Martin W. F., Muller M., 2007. Origin of Mitochondria and Hydrogenosomes. Heidelberg, De, Springer Verlag. 122 Emilianorf VV, April 2003. Mitochondrial connection to the origin of the eukaryotic cell. European Journal of Biochemistry. 270, 8 1599 1618 do, 10.1046/J.1033.2003.2003.03499.x PMID 126941744 123 Muller M., Martin W., May 1999. The Genome of Rickettsia Prowers-Key and Some Thoughts on the Origin of Mitochondria and Hydrogenosomes. Bioessays. 21, 5377381 do. 10.1002 /CSI 1521-1878, 199905, 21:53.0.co, 2w. PMID 103760009. 124 Gray M.W., Berger G., Lang B.F., March 1999. Mitochondrial Evolution. Science. 283, 5407 1476 1481. Bibcode 1999 SCI 283.1476G. Do 10.1126/science.283.5407.1476. PMC 34287767. PMID 100666161. 125 Thrash J.C., Boyder, Huggett M.J., Grota J., Karen I.P., Yoda R.J., it al. 14 June, 2011. Phylogenomic evidence for a common ancestor of mitochondria and the SAR11 clade. Scientific reports. 113. Bibcode 2011 Nat SR 1E13T. Do 10.1038/SREP00013. PMC 32165501. PMID 223555532. 126 Martigan J., Vosberg J., Guy L., Ofra P., Etima T. J., May 2018. Deep mitochondrial origin outside the sampled Alpha-Proteobacteria. Nature. 557, 7703 101105. Bibcode 2018 Nature.557101m. Do 10.1038/S41586-018-005-5. HDL 1874/373336. PMID 296958565. S2 SID 137400626. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 127 Fan L, Woody, Gormikin V, Xiao J, Zhu Y, Garges, it al. September 2020. Phylogenetic analyses with systematic taxon sampling show that mitochondria branch within Alpha-Proteobacteria. Nature, Ecology and Evolution. 4.9. Springer Science and Business Media LLC, 1213-1219. Bibcode 2020 NATE 4.1213F. Bioexif 10.1101/715870. Do 10.1038/S41559-020-1239. X. PMID 32661403. S2 SID 2205074552. 128 Carat Wang S, Luo H, June 2021. Dating Alpha Proteobacteria Evolution with Eukaryotic Fossils. Nature Communications. 12, 1. 3324 Bibcode, 2021 Natco 12.3324 Do. 10.1000.3467-021-23645-4. PMC 81757736. PMID 34083540. 129 Carat Espasti MD, Geiger, Sanchez Flores, the 16th of May 2022. On the bacterial ancestry of mitochondria, new insights with triangulated approaches. BioExiv, 2022.05.15.491939. Do. 10.1121/2022.05.15.491939. S2 SID 2488360055. 130 Carat Munoz Gomez Sa, Cisco E, Williamson K, Iml, Slamovitz CH, Moreira D, It al. March 2022. Site and branch. Heterogeneous analyses of an expanded dataset favor mitochondria as sister to known alpha proteobacteria, PDF. Nature. Ecology and Evolution. 6, 325362. Bibcode, 2022 NATE, 6253M. Do. 10.1038/S41559-021-01638-2. PMID 35027725. S2 SID 24595841. Archived, PDF, from the original on the 18th of November 2023. Retrieved the 30th of December 2023. 131 Carat Shonmi, Martigen J, Vosberg J, Kostelbacher S, Etima TJ, August 2022. The evolutionary origin of host association in the Rickettsia. Nature Microbiology. 7, 8 1189 1199 do. 10.1038/S41564-022-01169X. PMC 9352585. PMID 357988888. 132 Carat O'Brien TW, September 2003. Properties of human mitochondrial ribosomes. EUBM Life. 55, 9505513. Do. 10.1080/152165403100016266610. PMID 146585756. S2 SID 43415449. 133 Carat Sagan L, March 1967. On the Origin of Mitosing Cells. Journal of Theoretical Biology. 14, 325524. Bibcode, 1967 B 14225S. Do. 10.1016/0022-5193, 67, 90079-3. PMID 11541392. 134 Carat Emilianov VV, February 2001. Rickettsiaceae, rickettsia-like endosymbionts, and the origin of mitochondria. Bioscience. Reports. 21/1117 do. 10.1023/1010409409415723. PMID 11508688. S2 SID 101442020. 135 Feng DF, Choji, Doolittle RF, November 1997. Determining divergence times with a protein clock, update and revaluation. Proceedings of the National Academy of Sciences of the United States of America. 94, 241302813033. Bibcode 1997 NAS 9413028F. Do 10.1073/NAS.94.24.13028. PMC 24257. PMID 9371794. 136 Cavalier Smith T. 1991. Archamoeba, the ancestral eukaryotes. Biosystems. 25/12/2538. Bibcode 1991 BISIS 25/25C. Do 10.1016/0303-2647, 91, 90010I. PMID 1854912. 137 Danovaro R, Delano A, Pasteur, Gambi C, Heine I, Christensen R.M., April 2010. The first metazo living in permanently anoxic conditions. BMC Biology. 8:30. Do 10.1141-1741-7007-8-30. PMC 2907586. PMID 203700908. 138 Kokhalo, the 7th of April 2010. The mud creature that lives without oxygen. Zoologa. New Scientist. Archived from the original on the 26th of February 2020. Retrieved the 27th of February 2020. 139 Shifletum, Johnson P.J., 2010. Mitochondrion-related organelles in eukaryotic protists. Annual Review of Microbiology. 64. 409429. Do 10.101146./anav.micro.62.0813007.162826. PMC 3208401. PMID 205286887. 140 Kharkowska, Triatli SC, Psoo, Novak L, Varsik V, Sukal P, It al. October 2019. Batischazifu, ed. The oxymonad genome displays canonical eukaryotic complexity in the absence of a mitochondrion. Molecular Biology and Evolution. 36. 10.2292 do 10.1093/Molbev/MSZ147. PMC 6759080. PMID 313871118. Archived from the original on the 12th of April 2024. Retrieved the 14th of April 2024. 141 ABC Chan DC, June 2006. Mitochondria, dynamic organelles in disease, aging, and development. Cell. 125, 7 1241. 1252 do. 10.1016/j.cell.2006.06.010. PMID 168147112. S2 SID 8551160. 142 AB Wiesner RJ, Ruag JC, Morano I, March 1992. Counting target molecules by exponential polymerase chain reaction, copy. Number of mitochondrial DNA in rat tissues. Biochemical and Biophysical Research Communications. 183, 2553559. Do. 10.1016/0006-291X, 92, 905170. PMID 1550563. 143 Falkenberg M., July 2018. Mitochondrial DNA replication in mammalian cells, overview of the pathway. Essays in Biochemistry. 62, 3287296 10.1042/EBC20170100. PMC 6056714. PMID 2988072. 144 AB Anderson S., Bankier at, Barrel BG, De Bruijn MH, Coulson R., Druin J., it al. April 1981. Sequence and Organization of the Human Mitochondrial Genome. Nature. 290, 5806 457465. Bibcode, 1981 Nature.294570. Do. 10.1038/2904570. PMID 7219534. S2 SID 435557. 145 Fukuhara H., Saw F., Dressai R., Dienal N., Miyakawa I., Rasit S., it al. April 1993. Linear mitochondrial DNAs of yeasts. Frequency of occurrence and general features. Molecular and cellular biology. 13, 4 2309 2314. Do. 10.1028/MCB.13.4.2309. PMC 359551. PMID 8455612. 146 Bernardigi, December 1978. Intervening sequences in the mitochondrial genome. Nature. 276, 5688 558559. Bibcode, 1978 Nature.276558b. Do. 10.1038/2765580. PMID 214710. S2 SID 431438. 147 Hebar SK, Belcher SM, Perlman PS, April 1992. A maturase encoding group IIA intron of yeast mitochondria self-splices in vitro. Nucleic acids research. 20, 7 1747 1754 do. 10.1093//na/20.7.1747. PMC 312,266. PMID 1579468. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 148 Gray MW, Lang BF, Cedar R, Golding GB, Lemyo C, Sankoff D, et al. February 1998. Genome Structure and Gene Content in protist mitochondrial DNAs. Nucleic Acids Research 26, 4865878 10.1093/na/26.4.865. PMC 147,373. PMID 9461442. 149 Gray MW, Lang BF, Berger G, 2004. Mitochondria of Protists. Annual Review of Genetics. 38, 477524. Do. 10.100146/anav.genet.37.1108001.142526. PMID 155688984. 150 Telenis AG, Loha P, Kirino Y, Ragazos I, 2014. Nuclear and mitochondrial lookalikes in the human genome. Frontiers in Genetics. 5, 344. Do. 10.3089/fgene.2014.00344. PMC 4189335. PMID 2533997. 151 Shao R, Kirkis F, Barker SC, May 2009. The single mitochondrial chromosome typical of animals has evolved into 18 minochromosomes in the human body. Laus, Pediculus humanus. Genome Research 19, 59049112. Do. 10.1011/GR083188.108. PMC 2675979. PMID 193364551. 152 Castro J.A., Picornello, Raymond M., December 1998. Mitochondrial DNA, a tool for populational genetics studies. International Microbiology 1, 4327332. PMID 109433382. 153 Can RL, Stoneking M., Wilson AC, January 1987. Mitochondrial DNA and Human Evolution. Nature. 325, 6099 3136. Bibcode, 1987 Nature.325, 31c. Do. 10.1038/3250310. PMID 30257745. S2 SID 4285418. 154 Toronio, Achilleo, Macaulay V., Richards M., Bandelt H.J., June 2006. Harvesting the Fruit of the Human Mount DNA Tree. Trends in Genetics. 22, 6339345 do. 10.1016.j.tig.2006.04.001. PMID 1667830. 155 AB Garrigan D, Hammer MF, September 2006. Reconstructing Human Origins in the Genomic Era. Nature Reviews. Genetics. 7, 9669682, 10.1038/NRG 1941. PMID 1692135. S2 SID 176541. 156 Krings M, Stoner, Schmitz R.W., Kranitsky H., Stoneking M., Parbo S., July 1997. Neanderthal DNA Sequences and the Origin of Modern Humans. Cell. 90, 1932. 10.1032-S0092-8674, 00-80310-4HDL, 11858-/00001M0000002509608. PMID 9230299. S2 SID 13581775. 157 Source P, Ermini L, Thompson N, Moore M, Wright O T, Roller, it al. June 2009. Correcting for Purifying Selection, an Improved Human Mitochondrial Molecular Clock. American Journal of Human Genetics. 84, 674759. Do. 10.1016/J.H.2009.05.001. PMC 2694979. PMID 195007773. 158 Nachman M.W., Crow LSL, September 2000. Estimate of the mutation rate per nucleotide in humans. Genetics. 156, 1. 297304. Do. 10.1093/genetics/156.1.297. PMC 1461236. PMID 109789293. 159 Crick FH, Orgel, 1973. Directed Panspermia, PDF. Icarus. 19, 334136. Bibcode, 1973 ICAR, 19341C. Do. 10.1016/0019-1035, 73, 90110-3. Archived, PDF, from the original on 29 October 2011. Retrieved 21 October 2014. p. 344. It is a little surprising that organisms with somewhat different codes do not coexist. Further discussion archived. 3 September 2011, at the Wayback Machine. 160 Barrel BG, Bankia at, Druin J., November 1979. A different genetic code in human mitochondria. Nature. 282, 5735 189. 194 Bibcode, 1979 Nature.282189b. Do. 10.1038/2821890. PMID 226894. S2 SID 43358828. 161 ABL Zanowskio, Ostil J., 7 January 2019. The Genetic Codes. www.nkb.nlm.ni.gov. Archived from the original on the 13th of May 2011. Retrieved the 10th of February 2023. 162 Dukes Th. Osawa S. December 1990. The genetic code in mitochondria and chloroplasts. Experientia. 46, 11 12 1117. 1126 to 10.1007/BF0193691. PMID 2253709. S2 SID 1926496. 163 Hizela, Wissinger B., Scuster W., Brennico, December 1989. RNA editing in plant mitochondria. Science. 246, 4937. 1632 1634. Bibcode 1989 SCI 246.1632H. Do 10.1126/science.2480644. PMID 2480644. 164 Tabascal F. Posadar D. Knight Road, Zadoya R. May 2006. Parallel evolution of the genetic code in arthropod mitochondrial genomes. PLOS Biology. 4, 5E127 do 10.1337.journal.bio.0040127. PMC 1440934. PMID 166200150. 165 Pfeffer RF, 2012. Parkinson's disease. CRC Press. P 583. ISBN 978-1439807149. 166 Saoa, Josephum, Dutta D., Huang J.C., Aries J.P., Leeuwenberg C., August 2010. New insights into the role of mitochondria in aging, mitochondrial dynamics and more. Journal of Cell Science. 123, pt 15 2533 2542 do 10.1242/JCS.070490. PMC 2912461. PMID 209400129. 167 Hu GB, August 2014. Whole cell cryo-electron tomography suggests mitochondria divide by budding. Microscopy and microanalysis. 20, for 1180 1187. Bibcode 2014 mimic 20.1180H. Do 10.1017/S14319276140013117. PMID 248708111. S2 SID 74406669. 168 McWilliams TG, Suo Malanano, January 2019. Mitochondrial DNA can be inherited from fathers, not just mothers. Nature. 565, 7739-2962997. Bibcode 2019 Nature.565296M. Do 10.1038/D41586-019-00093-1. PMID 30643304. 169 Kimball, J.W. 2006, Sexual Reproduction in Humans, Copulation and Fertilization, archived 2 October, 2015, at the Wayback Machine, Kimball's Biology Pages, based on Biology, 6th ed., 1996. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 170 Sutovsky P. Moreno Road, Ramalho Santos J. Dominco T. Simile C. Scotton G. November, 1999. Ubiquitin tag for sperm. Mitochondria. Nature. 402. 6763 71372. Bibcode, 1999 Nature.402371s. Do, 10.1038/4646466. PMID 105868773. S2 SID 2050546671. Discussed in Science News, archived 19 December, 2007, at the Wayback Machine. 171 Mogensen HL, 1996. The Hows and Whys of Cytoplasmic Inheritance in Seed Plants. American Journal of Botany. 83, 3. 383404. Do. 10.2307/24467172. JSTA 2446172. 172 0Z, December, 2000. The exceptional mitochondrial DNA system of the muscle family Mytillidae. Genes and Genetic Systems. 75. 6313318 10.1266/GGS.75.313. PMID 11280005. 173 Sutherland B., Stuart D., Kensington 0Z, January 1998. The Fate of Paternal Mitochondrial DNA in Developing Female Muscles, Mytilus edulis, Implications for the Mechanism of Double Uniparental Inheritance of Mitochondrial DNA. Genetics. 148, 13413447 do, 10.1038/genetics/148.1.341. PMC 1459795. PMID 9475744. 174 male and female mitochondrial DNA lineages in the blue mussel, Mytilus edulis, species group, archived 18 May 2013. At the Wayback Machine by Donald T. Stewart, Carlos Saavedra, Rebecca R. Stanwood, Amy Zero. Ball, and Eleftherios Zouros. 175 Johns Dr., October 2003. Paternal transmission of mitochondrial DNA is, fortunately, rare. Annals of Neurology. 54, 4422. 424 do, 10.1002/Anna.107771. PMID 145206551. S2 SID 90422. 176 Thiagarajan B., Padura, Campbell C., November 1996. Mammalian mitochondria possess homologous DNA recombination. Activity. The Journal of Biological Chemistry. 271, 442753627543 do, 10.1074/JBC.271.44.27536. PMID 891039. 177 Lund DH, Hyman BC, May 1997. Animal mitochondrial DNA recombination. Nature. 387, 6630 247. Bibcode, 1997 Nature.387247L. Do. 10.1038/387247A0. PMID 915338. S2 SID 4318161. 178 Airwalker, Smith NH, Smith JM, March 1999. How clonal are human mitochondria? Proceedings. Biological Sciences. 266, 1418 477483 do. 10.1098/RSPB.1999.0662. PMC 1689787. PMID 101897111. 179 Tawadala P. Airwalker, Smith JM, December 1999. Linkage disequilibrium and recombination in hominid mitochondrial DNA. Science. 286, 5449 2524 2525 do. 10.1026/science.286.5449.2524. PMID 106174771. S2 SID 276852885. 180 Cree LM, Samuels DC, D'Souza Lopes SC, Rajasim HK. 1 Apinage P. Mann Jr., et al. February 2008. A reduction of mitochondrial DNA molecules during embryogenesis explains the rapid segregation of genotypes. Nature Genetics. 40, 2249. 254 do. 10.1038/2007.63. PMID 182236551. S2 SID 205344980. 181 Cow L, Shitara H, Hurtu T, Nagal Y, Imai H, Abe K, et al. March 2007. The mitochondrial bottleneck occurs without reduction of mount DNA content in female mouse germ cells. Nature Genetics. 39, 3386390 do. 10.1038/1970. PMID 172938666. S2 SID 106866347. 182 YT, Tilly D, Showbridge EA, December 2008. The mitochondrial DNA genetic bottleneck results from replication of a superpopulation of genomes. Nature Genetics. 40, 12 1484 1488 do. 10.1038/258. PMID 1902991. S2 SID 225349. 183 Johnston IG, Bergstaller JP, Have Lysik V, Kolb T, Rulik T, Brim G, et al. June 2015. Stochastic modeling, Bayesian inference, and new in vivo measurements elucidate the debated Mount DNA bottleneck mechanism. eLife 4, E07464. R14, 1512.02988. Do 10.7554/elife.07464. PMC 44868817. PMID 260355426. 184 Greedler R. Garm C. Steffensner T. 2012. Nuclear and mitochondrial DNA repair in selected eukaryotic aging model systems. Oxidative medicine and cellular longevity. 2012, 282,438. Do 10.1155/2012/2822438. PMC 3462412. PMID 230500036. 185 ABC Saki M. Prakasha, June 2017. DNA damage-related crosstalk between the nucleus and mitochondria. Free radical. Biology and Medicine. 107, 216227. Do 10.1016/j.freered-by-2016.11.050. PMC 5449269. PMID 279150046. 186 Leon J. Sakumi K. Castillo E. Xing Z. Oka S. Nakabapu Y. February 2016. 8-oxyguanine accumulation in mitochondrial DNA causes mitochondrial dysfunction and impairs neuritogenesis in cultured adult mouse cortical neurons under oxidative conditions. Scientific reports. 6, 22086. Bib code 2016 NAT SR 622086L. Do 10.1038/SREP22086. PMC 4766534. PMID 2691270. 187 Dahal S. Dubey S. Raghavan SC, May 2018. Homologous recombination-mediated repair of DNA double-strand breaks. Operates in mammalian mitochondria. Cellular and Molecular Life Sciences. 75, 9 1641 1655 do 10.1074/S00018-017-2702. Y. PMC 11105789. PMID 291163662. S2 SID 477960. 188 Odahara M. Inuyiti, Fujitati, Haspem, Sikine Y. February 2007. Involvement of mitochondrial targeted REC-A in the repair of mitochondrial DNA in the MOS, Fisca-Mitrella patterns, genes and genetic systems. 82, 14351 do 10.1066/GGS.82.43. PMID 1739619. 189 Tadi SK, Sebastian R. Dahal S. Babu RK, Chowdhury B. Raghavan SC, January 2016. Microhomology-mediated end joining is the principal mediator of double-strand break repair during mitochondrial DNA lesions. Molecular biology of the cell. 27, 2. 223235. Do 10.1091/MBC.E15-05-0260. PMC 4713127. PMID 266097070. 190 AB Henriquez FL, Richards Ta, Roberts F, McLeod R, Roberts CW, February 2005. The unusual mitochondrial compartment of Cryptosporidium parvum. Trends in Parasitology. 21, 26874 do. 10.1016/J.PT 2004.11.010. PMID 1566459. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 191 John U, Lu Y, Walrab S, Groth M, Janukovic J, Kohli G.S., it al. April 2019. Anaerobic eukaryotic parasite with functional mitochondria that likely lacks a mitochondrial genome. Science Advances. 5, 4 Av 1110. Bibcode 2019 SCIA 5.1110J. Do 10.1126/Seadv.Av 1110. PMC 6482013. PMID 31032404. 192 Veritable powerhouse even without DNA. Parasitic algae from the dinoflagellate lineage have organized their genetic material in an unprecedented way. Science Daily. Archived from the original on 24 June 2019. Retrieved 8 May 2019. 193 ABC Ziviani M. Diidonato S. October 2004. Mitochondrial disorders. Brain. 127. PT 10 2153 2172. Do 10.1093/brain/or259. PMID 153586637. 194 Taylor R.W., Turnbull D.M., May 2005. Mitochondrial DNA mutations in human disease. Nature Reviews. Genetics. 6, 5389. 402 do. 10.1038/NRG 1606. PMC 1762815. PMID 1586121. 195 Goldman O, Khist S, Freeinkman E, Dewino, Majumder B, Mundell J, it al. August 2019. Targeting tumor phenotypic plasticity and metabolic remodeling in adaptive cross-drug tolerance. Science Signaling. 12, 595. Do 10.1126/Signal.AAs 8779. PMC 7261372. PMID 31431543. 196 Sahati, -C, Jabalan R, Khist S, Kukanio, Kurmi K, it al. January 2022. Intercellular nanotubes mediate mitochondrial trafficking between cancer and immune cells. Nature Nanotechnology. 17, 198106. Bibcode 2022 NatNa 17, 98S. Do 10.1038//41565-021-01000-4. PMC 10071558. PMID 34795441. S2 SID 2443498825. 197 China EPF, Shonier, September 2003. Mitochondria. Journal of Neurology, Neurosurgery, and Psychiatry. 74, 9 1188. 1199 do. 10.1136/JNNP.74.9.1188. PMC 1738655. PMID 129333917. 198 Shia TB, Beta Beta, Greenemeier JT, June 2002. Environment, Mitochondria, and Parkinson's disease. The Neuroscientist. 8, 31921997 do. 10.101388584020080030030004. PMID 120614998. S2 SID 24318009. 199 Gomez C., Bandes MJ, Navarro, January 2007. Pesticides and impairment of mitochondrial function in relation with the Parkinsonian syndrome. Frontiers in Bioscience. 12, 1079 1093. Do. 10.2741/2128. PMID 171273363. 200 Lim Ya, Rhine V, Basang G, Maya F, Poljako, Rafter EMJ, et al. April 2010. A beta and human amylin share a common toxicity pathway via mitochondrial dysfunction. Proteomics. 10, 8 1621 1633 do. 10.1002/PMIC.2009000651. PMID 201867753. S2 SID 33077667. 201 King JV, Liang WG, Skera Pez KP, Schilling AB, Meredith SC, Tang WJ, July 2014. Molecular basis of substrate recognition and degradation by human pre-sequence protease. Structure. 22, 7996 1007 do. 10.1016/J. Street 2014.05.003. PMC 412888. PMID 2493149. 202 Skapira R, July 2006. Mitochondrial disease. Lancet. 368, 9529 7082 do. 10.1016/S0140-6736, 06, 68970-8. PMID 168153881. S2 SID 27039617. 203 Pechenic SR, Newstat J, August 2007. Mitochondrial dysfunction and molecular pathways of disease. Experimental and molecular pathology. 83, 18492 do. 10.1016/J.Yexmp.2006.09.008. PMID 1723937. 204 Bugger H, Abel ed., November 2010. Mitochondria in the diabetic heart. Cardiovascular research. 88, 2229240. Do. 10.1093/CVR/CVQ239. PMC 2952534. PMID 206392113. 205 Podolaka, Waklawik Potakai, Lukasik K., February 2022. The role of mitochondria in human fertility and early embryo development. What can we learn for clinical application of assessing and improving mitochondrial DNA cells? 11, 5797. Do. 10.3390/cells 110507797. PMC 8909547. PMID 35269419. 206 Maypan Laub P., Bognet M., Hakem He, Buat Pei, Rainier P., January 2021. Embryo and its mitochondria. Antioxidants. 10, 139 Do. 10.3394/Antiox 100200139. PMC 7908991. PMID 33498182. 207 Committee on the Ethical and Social Policy Considerations of Novel Techniques for Prevention of Maternal Transmission of Mitochondrial DNA Diseases, Board on Health Sciences Policy, Institute of Medicine, National Academies of Sciences, Engineering, and Medicine, the 17th of March, 2016, Claiborne, English R. Khan J., eds, Introduction, Mitochondrial Replacement. Techniques, Ethical, Social, and Policy Considerations, National Academies Press, U.S., retrieved 5 December, 2023. 208 Richter C., Park J.W., Ames B.N., September, 1988. Normal oxidative damage to mitochondrial and nuclear DNA is extensive. Proceedings of the National Academy of Sciences of the United States of America. 85, 17 6465 6467. Bibcode, 1988 NAS, 85.6465 R. Do. 10.1033/NAS.85.17.6465. PMC 281993. PMID 34131108. 209 Harmon D., July 1956. Aging, a theory based on free radical and radiation chemistry. Journal of Gerontology. 11, 3298300. Site CX 10.1.1.663.3809. Do. 10.1093/Geronge/11.3.298. PMID 133322224. 210 Harmon D., April 1972. The Biologic Clock, the Mitochondria. Journal of the American Geriatrics Society. 20, 414547. Do. 10.1512/J.1052-5432-5472.TB00787.X. PMID 5016631. S2 SID 396830. 211 Mitochondria and Aging. CircuitBlue.co. Archived from the original on the 29th of September 2017. Retrieved the 23rd of October 2006. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 212 Bofoli D, Skako SC, Vergaria, Celerino G, Santa Cruz G, Papa S, April 1994. Decline with age of the respiratory chain. Activity in human skeletal muscle. Biochimica et Biophysica Acta, BBA, molecular basis of disease. 1226, 17382. Do. 10.1016/0925-4439, 94, 90061-2. PMID 8155742. 213 de Gray AD, 2004. Mitochondrial mutations in mammalian aging, an over-hasty about turn. Rejuvenation Research. 7, 3. 171174. Do. 10.1089/ridge.2004.7.171. PMID 155888517. 214 Bender, Krishnan KJ, Morris cm, Taylor GA, Rive K, Perry RH, et al. May 2006. High levels of mitochondrial DNA. Deletions in substantia nigra neurons in aging and Parkinson disease. Nature Genetics. 38, 5515517 do. 10.1038/1769. PMID 166040774. S2 SID 1395698. 215 Meta R, Walters R, Waldron FM, Pal S, Selvaroj BT, McLeod Mr., et al. August 2019. Targeting mitochondrial dysfunction. In amyotrophic lateral sclerosis, a systematic review and meta-analysis. Brain Communications. 1 FCZ009. Do. 10.1093/braincoms/FCZ009. PMC 7056361. PMID 321335457. S2 SID 2011686692. 216 Meta R, Gregory J.M., Dando O, Carter R.N., Burke, Nanda J., et al. February 2021. Mitochondrial bioenergetic deficits in C9 of 72 amyotrophic lateral sclerosis. Motor neurons cause dysfunctional axonal homeostasis. Acta neuropathologica. 141, 2. 257279. Do. 10.1074/S00401-020-02252-5. PMC 7847443. PMID 333984003. 217 Jiang JL, Shukla P, Pajadas K, Ahmed NS, Kari S, Gundi D, et al. November 2020. Mitochondria in ovarian aging and reproductive longevity. Aging Research Reviews. 63, 101,168. Do. 10.1016/J.2020.1011168. PMC 9375691. PMID 328966666. S2 SID 2214727721. 218 Colica, 1857. Einig bemerken über die Entdeckungen der Hautnerven und den Bau der Muskel, some remarks. About the terminations of the cutaneous nerves and the structure of muscles. Zeitschrift für Wissenschaftliche Zoologie, in German. 8, 311325. Archived from the original on the 22nd of June, 2023. Retrieved the 22nd of June, 2023. On p. 316, Colica described mitochondria which he observed in fresh frog mussels, see a blasrund like Kanchin, Welsh in Langen Linien Fortmigen. Zuzhang, wenn man einmal auf Diesel ben aufmerksam geworden ist. They are very faint round granules, which are embedded in the muscles, contractile substance in long linear trains. These granules are located in the whole thickness of the muscle fiber, on the surface as in the interior, and they are so numerous that they appear as a not unimportant element of the muscle fibers, once one has become alert to them. Colica said, p. 321, that he had found mitochondria in the muscles of other animals. In Figure 3 of Table 14 archived the 22nd of June, 2023, at the Wayback Machine, Colica depicted mitochondria in frog muscles. 219 A B C D E F G H I J Ernst L. Scots G. December 1981. Mitochondria, a Historical Review. The Journal of Cell Biology. 91, 3 pt. 2227S255S. Do. 10.1083/JCB.91.3.227S. PMC 2112799. PMID 7033239. 220 Altman R. 1890. Die Elementarorganismen und ihre Beziehungen zu den Zellen, Elementary Organisms and their Relations to Cells, in German. Leipzig, Germany, Veit and Company p. 125. Archived from the original on the 23rd of June, 2023. Retrieved the 23rd of June, 2023. From p. 125, the Orch sonst Machelei Umstände des Versprechen, das Mikroorganismen und Granule einander gleichwertig sind und Elementarorganismen vorstellen, welche sich überall finden, wo lebendige Kraft ausgelost wurden, so wollen wir sie mit dem gemeinschaftlichen Namen der Bioblasen bezeichnen. Since otherwise some circumstances indicate that microorganisms and granula are equivalent to each other and suggest elementary organisms, which are to be found wherever living forces are unleashed, we will designate them with the collective name of bioblasts. 221 Mitochondria. Online Etymology Dictionary. Archived from the original on the 4th of March, 2016. Retrieved the 23rd of May, 2013. 222 Bender C., 1898. Über die Spermatogenese der Vertebraten und höhere Evertebraten. 2. Thiel, die Histogenese der Spermien, on spermatogenesis in vertebrates and higher invertebrates. Part 2, the histogenesis of sperm. Archive für Physiology, in German. 1898, 393398. Archived from the original on the 24th of February, 2019. Retrieved the 14th of January, 2018. From p. 397, after Brenda states that ich bereits in vielen Zelaten alle möglichen Klassen gefunden habe. I have already found them, mitochondria, in many types of cells of all possible classes of animals. He suggests, ich mocht vorläufig vorschlagen, einen als Mitochondria Einbesondstellung vorzubehalten, die ich in Wittenarbeiten begründen. Word. I would like to suggest provisionally reserving for them, as mitochondria, a special status which I will justify in further work. Bender C., 1899. Whittier Mitteilungen über die Mitochondria, further reports on mitochondria. Archiv für Physiologie. Verhandlungen der Berliner Physiologischen Gesellschaft, in German. 1899, 376383. Archived from the original on June 23, 2023. Retrieved the 23rd of June, 2023. 223 Michaelis L., 1900. Die Vitalfarbung, ein Darstellungsmethode der Zellgranula, the vital stain, a method of imaging cell granules. Archiv für mikroskopische Anatomie und Entwicklungsgeschichte, Archive for Microscopic Anatomy and Ontogenesis. In German. 55, 558575. Do. 10.1001/BF02977747. S2 SID 8501030. Archived from the original on the 23rd of June, 2023. Retrieved the 23rd of June, 2023. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 224 Ernster citation Mivs F., May 1908. Die Chondriosomen als Trager Erblichen Lagen. Cytologisch Studienem. Hunier Embryo. Archiv für mikroskopische Anatomie. 72, 1816867 do, 10.1007/BF02982402. S2 SID 850473330. Archived from the original on the 9th of January, 2020. Retrieved the 24th of June, 2019. Is wrong, correct citation is Mivs F., 1904. Über das Vorkommen von Mitochondrien bis Chondromiten in Pflanzenzellen. B. DTSCH. Bot. Giz. 22, 284286. Do. 10.1111/J.1438. 8677.1904.TB05237.X. S2 SID 257822147, cited in Mivs 1908 paper and in Schmidt U. 1913. Flans like Mitochondrion. Progressus Rei Botanici. 4, 164183. Retrieved the 21st of September, 2012, with confirmation of Nymphea alba. 225 Kingsbury B.F., 1912. Cytoplasmic fixation. The anatomical record. 6, 23952 do, 10.1002/R.1090060202. S2 SID 2214000422. Archived from the original on the 23rd of June, 2023. Retrieved the 23rd of June, 2023. From P47, the mitochondria are the structural expression thereof, i.e., of the chemical reducing processes in the cytoplasm. 226 Warburgo, 1913. Über Sauerstoffet Mendikanchine US Lebertellen und über Sauerstoffetmang in Birkefeld filtraten. Wasriger Leberextrake, on respiring granules from liver cells and on respiration in Birkefeld filtrates of aqueous liver. Extracts. Pflügers Archiv für Digest Physiologie des Menschen und der Tier, Pflügers Archive for all physiology of humans and animals, in German. 154, 599617. Archived from the original on 23 June, 2023. Retrieved 23 June, 2023. 227 Millen L, Trive DM, Singer, Talika M, Amigi M, June, 2015. Mitochondrial Biology, Targets, and Drug Delivery. Journal of Controlled Release. 207, 4058. Do. 10.1016/j.general.2015.03.036. PMID 258416999. 228 Martin W.F., Garges, Z. Muscovy, September 2015. Endosymbiotic Theories for Eukaryote Origin. Philosophical Transactions. Of the Royal Society of London. Series B, Biological Sciences. 370, 1678 20140330. 10.1098/rstb.2014.0330. PMC 4571569. PMID 263237761. General. This article incorporates public domain material from Science Primer. NCBI. Archived from the original on 8 December 2009. External links. Lane N. 2016. The Vital Question, Energy, Evolution, and the Origins of Complex Life. W. W. Norton and Company. ISBN 978-0393352979. Powering the Cell Mitochondria, archived 17 August, 2022, at the Wayback Machine Xvivo Scientific Animation. Mitod.com, archived 3 July, 2013, at the Wayback Machine, the Mitochondrial Disease Database. Mitochondria Atlas, archived 29 June, 2012, at the Wayback Machine at University of Mainz. Mitochondria Research Portal at mitochondrial.net. Mitochondria, Architecture Dictates Function at cytochemistry.net. Mitochondria links at University of Alabama. MIP, archived 23 May, 2016, at the Portuguese Web Archive Mitochondrial Physiology Society. 3D Structures of Proteins from Inner Mitochondrial Membrane, archived 13 September, 2011, at the Wayback Machine at University of Michigan. 3D Structures of Proteins Associated with Outer Mitochondrial Membrane, archived 13 September, 2011, at the Wayback Machine at University of Michigan. Mitochondrial Protein Partnership at University of Wisconsin. Mitominer, a mitochondrial proteomics database, permanent dead link, at MRC Mitochondrial Biology Unit. Mitochondrion Cell-Centered Database, archived 7 October, 2011, at the Wayback Machine. Mitochondrion Reconstructed by Electron Tomography, archived 14 November, 2012. At the Wayback Machine at San Diego State University. Video clip of rat liver mitochondrion from cryo-electron tomography. Categories: cellular respiration, endosymbiotic events, mitochondria. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.